I think we can go ahead and get started at we'll start with Valerie and Adam, I think, kicking the day off, and then get into some of the work that folks have been doing on the cloud platforms. So over to you, Valerie.
Awesome. David, you can go ahead to the next slide. So, allow me to be the second person to say welcome. This is our second virtual NCPI workshop, but our third total NIH Cloud Platforms Interoperability workshop. So these four platforms started working together about a year ago. You all have made some really good progress over the course of the year. And I also want to acknowledge that the process of defining what you are doing and sharing that information with the public is not so trivial. So congratulations on going public and launching this web resource, which describes our NCPI activities. And of course, thanks to the Office of Data Science Strategy for hosting this information on their website. Next slide. Since the Kids First DRC is hosting today's workshop, I just want to share some perspective on the science that is driving the overall NIH Kids First program and the development of the Kids First data resource, as well as our eagerness for interoperability. Kids First is a trans-NIH Common Fund program, and we're focused on the genetic discovery of childhood cancers and structural birth defects and the potential intersection of these conditions. So cross-data set interoperability is inherent to the vision of Kids First, fostering research in these what tend to be rare conditions. Next slide. So both the challenge and the opportunity for us has been the vast diversity of conditions and phenotypes represented in the Kids First cohorts that we are actually still sequencing. And the fact that these are often subsets of larger projects for which data might live somewhere else. Next slide. Despite this challenge, thus far, the Kids First Data Resource Center has designed a system that not only enables the creation of synthetic cohorts based on queries of de-identified and clinical phenotypic data from Kids First data sets in the portal, but we already have interoperability with a few NCI target data sets as well. So in this case, the user is building a synthetic cohort of Kids First congenital heart disease data and neuroblastoma data from both Kids First and NCI, and then pushes these files into the cloud workspace, which is, in this case, 7 Bridges Cavatica, for combined analysis. And this is what we're really trying to enable across NCPI platforms. Next slide. But of course, particularly for us, we are starting with the childhood cancer and structural birth defects data sets that reside in Anvil, Biodata Catalyst, and the CRDC, and are of interest to the Kids First community. Next slide. But Kids First leadership is very interested in a future vision of federating additional pediatric data or Kids First relevant data sets to the Kids First data resource. So we're in the early stages of evaluating these additional opportunities as well. Next slide. But as you all know, supporting these use cases requires addressing multiple layers of interoperability. And these 4 working groups have spent the last year tackling these challenges, including addressing operational barriers, testing and implementing technical standards for data exchange, coordinating training resources for transitioning researchers to the cloud, and piloting FHIR standards for structuring and exchanging clinical and phenotypic data. And of course, today we'll hear about everyone's progress and the goals for the next coming year. Next slide. And this is by no means a comprehensive or directive list, but I did want to share or call out some additional pain points that have come up for us that may inform our roadmap discussion a little bit later in the day. So several of us are realizing that many researchers are hesitant to use the cloud if their favorite tools or workflows are not optimized for the cloud environments that we're using. So perhaps this is an opportunity for a new working group, or could be addressed by one or more of our existing working groups. There are additional data sets or platforms that we may want to interoperate with, but we are not yet sure how to engage them in NCPI or when the right time to engage them would be. There's a whole session on Monday devoted to how to estimate or manage cloud costs, not only for the platforms, but this is particularly important for engaging end users as well. And through the efforts of the FHIR working group, I think in the next year, we'll start to see a role for FHIR resources in creating a flexible framework for clinical data interoperability, even for data that are not derived from EHR, and for data that do not conform to existing common data models, which is very true for Kids First data sets that are so diverse. So with that, I will hand it over to Adam Resnick from the Children's Hospital of Philadelphia, who serves as the PI of the Kids First Data Resource Center.
Thanks, Valerie. So I'll only take a couple minutes, and it's really a pleasure for the Kids First DRC to host this third interoperability meeting. It may be because of time and COVID and political landscape, but I think probably all of us are more existentially oriented than usual in thinking about what we're doing. And so I was trying to think about the introduction for today. I really started recognizing some things and themes that have come up within the Kids First DRC as we began engaging both with you, but with many other stakeholders. And I just wanted to go through a couple of slides to test this framing moving forward. So when I talked about interoperability with different stakeholders, it turns out that people talk about interoperability towards different ends. And it turns out, like, even if you I just did this morning, Google interoperability, you get two different definitions. The first one, which is, I was surprised, is the interoperability between devices made by different manufacturers or for computers. Then the second one, right below it, is the ability of military equipment or groups to operate in conjunction with each other. And you can tell that the sort of to what ends question matters depending on whether or not you have different purposes for interoperability, whether it's building military complexes or interoperable systems. Next slide. Being in a children's hospital, and as Valerie pointed to, within the DRC, we constantly face questions about interoperability. And I have begun, and we have begun to really think about interoperability within this context. And so I showed this email from Mark Colley from Australia a few weeks ago. Next slide. Where he had reached out to me, with this following question. He's really a talented bioinformatician who is running a clinical trial where they're sequencing essentially different types of cancers in children, trying to leverage existing available data sets in real time in order to support decision-making. Next slide. And so I'm just going to go through the way that he thinks about it in this image and connect it to, I think, hopefully what will emerge in the following talks today and on Monday. So the first thing he notes is that the histopathology, the slide images that are available for these patients are especially challenging for the human pathologist to really interpret. Next slide. And that even the molecular landscape is not obvious in terms of pathognomonic mutations that classify the tumor. Next slide. He knows that this tumor resides in a certain location in the brain, and location oftentimes goes along with the prognosis and type of tumor, but the location doesn't quite match the mutation or the pathology slide. Next. He knows that the tumor has a canonical BRAF mutation that all of cancer actually has learned from since maybe almost 10 years ago. BRAF V600E mutations became targetable in the context of melanoma. So he can immediately harness pre-existing research on melanoma in the context of a pediatric brain tumor, but that's not the only mutations he finds. He also finds an H3F3B variant that actually seems to have a mutation that when it occurs in the A context of the gene actually describes a bone tumor, not a brain tumor. And next slide. And he can sort of reference this difference, and he doesn't know what it means. And so he goes to the literature, which is the next slide, and just recognizes that this is the one thing that he is sure about, is that the methylation status of this tumor correlates with the A version. And so he's sort of stuck in this context, and he's reaching out to me in the hope that I can help him, because what's going to happen is, depending on whether or not he classifies this tumor as a high-grade or a low-grade tumor, the treatment paradigm will change. And if it's a high-grade tumor, they'll be more aggressive. They'll give this child radiation therapy, likely, and they will risk the consequences of that therapy for the patient in order to try to save this patient's life. If it's a low-grade tumor, they'll try to mitigate the harm of the therapy and be less aggressive. Next slide. And so when I read that email, for me, Mark is trying to navigate these two realms. He's trying to navigate a realm of, what's the farmer's almanac, meaning sort of a knowledge base of pre-existing knowledge that I can connect my current state or predict this patient's tumor to really weather prediction for any particular day's weather or hurricane. I think our hurricane just passed us. And those are two very different activities, but in many ways, both are required. And one of the things that we've been iterating on within Kids First is really this combination of activities. Next slide. Another version of this is sort of the stock market and whether or not you have your mutual funds or 401k, or whether or not you're trading on a daily basis and trying to respond to real-time data. Next slide. And so we really feel that if you want to do that, if you want to be able to help Mark, you actually have to misspell FAIR and really begin with interoperability, because only if Mark can connect the dots across those elements in ways that go beyond the almanac version of data can he actually support that. And that turns out to be a recurrent feature of almost all biology, is that if you try to apply precision-based approach at the level of the individual, you have to think about different ways of connecting data sets. Next slide. And so it really comes to this question of thinking about data and interoperability in two frames, at least for us. What data and how do you get access to the data, or how you use data, but more importantly, where is the data, where is it being generated, and how does it arrive, and when does it arrive to the right constituents? Next slide. For Mark to actually get his answer today, this is what he had to do. Actually, he couldn't do this, and it's only because he connected with me, and by chance, I answered my email in a timely manner that he actually was able to do this. And Mark actually is leveraging a Cavatica instance in Australia that can leverage and connect to the Kids First data resource. He could search for those mutations and alterations in a pediatric brain tumor cohort in PCBio portal. But that version of PCBio portal doesn't actually contain the patient that he needs. The patient that he needs is actually stored in AACR Project Genie, and that data is actually in Synapse. And there are 2 patients out of 80,000 that have that specific mutation that actually suggest that this mutation is a high-grade tumor variant. To navigate all of this independently of interoperability and access and connectivity is almost impossible. And so in many ways, I hope that the framing of the workshop over the next 2 days really starts delineating sort of to what ends we are talking about interoperability, and this difference between the flow of data and the capacity to generate static and or almanac-like data sets. And the two are actually required to achieve our purpose, but actually have slightly different means and ends to them. And if we get it right, I think Mark and others will be successful in the future. Next slide. And so my hope is that, and I always ask this question of our team, it's like, how do we know if we're successful when we do something? And the reality is that you're successful when Mark doesn't actually have to recognize that he's interoperating. So that's another metric that I would sort of challenge us to reach, is that interoperability really means users not knowing that they're actually interoperating. And that's really the privilege and excitement I have about working with all of you on interoperability, is to sort of become anonymous, nameless, and non-existent. Last slide, I think. Yep. So hopefully it's fun and enjoyable and fruitful. And like Allison mentioned, we really hope that it is as interactive and engaging as possible.
Great. Thanks, Adam and Valerie, for kicking us off. And I think we'll have a couple interesting talks of the early research adopters who do know that they're interoperating and are helping us get to that
Right.
point. We'll kick it off with Harrison Brand, I think.
Hi, thanks. If you flip to the next slide, I actually have like a little introduction about myself. Thanks. Yeah, so I'm Harrison Brand. I'm currently an assistant professor in the Department of Neurology at Mass General Hospital, Harvard Medical School. I did my PhD at the University of Pittsburgh in more statistical genetics with Eleanor Feingold and Brenda Diergaard. I then went to MGH in the Center of Genomic Medicine, and I did a postdoc with Dr. Michael Telkowski. And really, what I focus on now and what I focus on in my postdoc is, the impact of structural variation in whole genome sequencing. And I've applied that across, a lot of different disease cohorts, including many that are, involved in multiple of the systems that are here today. So next slide. Yeah, perfect. So, you kind of may wonder why I was asked to do this talk, and I feel like, I have experience working with multiple of the kind of different groups here today. So I am a NIH Cohort 1 Fellow, where I've been working on SVs and a type 2 diabetes and glycemic trait data set. So I've been working with the Biodata Catalyst group here, which has been a great experience. I'm also a member of the Broad CCDG and CMG teams, which use the Anvil system from NHGRI. And so I'm very familiar with, grabbing data from that and analyzing data, with I guess for that we do, it's in Google Cloud. So we use Terra, the Terra system from the Broad for analyzing data from that. I also have been working as part of the Broad GMKF sequencing analysis team. And, as part of that with several of the different GMKF disease specific groups. And so I do have experience working with the Kids First Data Resource Center. And then, not really included in this group, but, important to my cloud experience, I've been working, when I was a postdoc, I worked with the Simon Simplex collection and the Nomad SV cohort, or the Nomad database cohort. Next slide. So let me just give a quick background on kind of structural variants in my research and what I do, and then I'll discuss kind of my interaction with the cloud and how some of my experiences. So there are 4 basic classes of structural variation in human genome: deletions, which are loss of DNA; duplications, which are gains of DNA; inversions, which is a flip of a DNA strand; and insertions, translocations, that are insertions of a new piece of DNA and somewhere into the genome. And if you combine these 4 different classes together, you can actually get complex events that are kind of different combinations of these events. Okay. When we're talking about SV discovery, using whole genome sequencing, there's kind of 5 different methods that you can use. Different algorithms use different approaches. No kind of algorithm uses all approaches. And these are just, the kind of different signals that we can find in whole genome sequencing for discovering SV. And by the way, if anyone has any questions, feel free to just ask. One of the problems that we've really had in the SV field is that there's not really any kind of like set method for analyzing SV with whole genome sequencing. And part of the problem is that, first off, this kind of balance of sensitivity versus specificity and this kind of filtering has been really challenging for the field. So, some programs call a lot of variants, but, there's a lot of false positives. Other programs, call lots of really high quality variants, but they miss a lot of variants. And so trying to kind of find that balance has been really challenging. And so, to overcome this, we decided to develop an ensemble SV program. If you go to the next slide. So this is kind of our way around this. And basically what we do is we grab a lot of different programs and run an ensemble SV pipeline. And that way we're trying to kind of take the strengths of all the different programs, but then we can kind of control maybe some noise by filtering out variants that are of lower quality. And so this has been designed in a module kind of framework. And so you can see that there's 8 different modules. And, each kind of module is designed so you can, we're constantly updating this. So this is by no means like a complete or finished product. So this is constantly being updated to work. And, we've really designed it to be flexible so you can really, as better algorithms come out, as better programs come out, we can really, flip things in and out. And so I'm just going to quickly run through this. So the first thing we do is we run several unfiltered algorithms to maximize sensitivity. Okay, next slide. We then use the kind of evidence from the BAM to evaluate whether we think that those variants are real or not to improve our specificity. And we run genotyping, and then we're able to capture both bounce and unbounce events. And then we have a step where we actually integrate SV signatures together to find complex events. And then there's this overall modular design. So we can easily add new pieces, and we can update things that we think need to be improved. Okay. So let me just talk about my actual experience in the cloud now. So my first experience probably started, it's probably been over 5 years now. The Simons Foundation for Autism Research in their Simons Simplex collection released these initial pilot data from 40 autism spectrum disorder families. And so I'm not sure how many of you are familiar with the Simons Foundation, but these are quartet families. So they have an affected proband with a healthy sibling and 2 unaffected parents. And so that gave us a total of 160 genomes. And this was, many years ago. So this was not, this was a lot of data for us. And, we hadn't really been working too much with whole genome sequencing before this, besides, maybe 1 or 2 genomes. Now all this data was hosted on AWS. And, at this point, we were very familiar with working with HPC. We had not worked very much in the cloud. And so what we did is we just pulled down the BAMS, our local computing cluster, which was about 16 terabytes of data, ran our SV detection locally, and then, deleted the BAMS, which we were able to do. But, we realized that as we were getting more and more samples on the cloud, and more genomes analyzed, it wasn't scalable. And we weren't going to be able to always pull these down and, keep analyzing these on local computing clusters. So we were going to have to start, doing work on the cloud. So next slide. So we started with kind of a hybrid approach. And so the Simons Foundation released a larger cohort, which was 519 families, which was, over 2,000 samples. And so this was certainly something that we couldn't just pull down to our local compute cluster and analyze. And so what we ended up doing was we ended up running the raw SV algorithms on AWS, and then taking those raw output and pulling those down, which was much more manageable, to our local computing cluster. And then, we were able to run our SV pipeline on the local compute cluster with those. It looks like, is there a question in chat? Oh, sorry. So there's a question, while calling the variants was being used as reference. And so it's just, you're aligning against the genome. And so you're just aligning against the reference genome. And so it's really, the reference is the, HG38 reference genome or HG19 reference genome. And you're really just comparing for differences between those. So you're looking for differences between the reference and your whole genome sequencing sample. Okay, sorry. So we could go, yeah, okay, perfect. So as we were running this, the hybrid approach worked, but we realized that this, as we keep expanding, this is really not going to scale. And at the same time, we also realized that, there was a need to create a kind of a larger population scale SV reference, because we were discovering SV in these cohorts, but we didn't really have anything to compare them against, because, no one had really done this to scale on large scale whole genome sequencing yet. Similar to something like that had been done with like EXAC or NOMAD for the, for exomes or whole genome sequencings respectively, for, smaller variants for SNVs and indels. This really hadn't existed for SVs beyond like a small 1,000 genome study that was done on 4 to 8x whole genome sequencing. And so we really wanted to kind of rectify this. So next slide. So we ended up running our SV pipeline on Illumina whole genome sequencing on about 15,000 samples. And this was a diverse set, and this was all done through the NOMAD cohort. And working with this, we realized really how important it is to, well, we learned several things. So 15,000 samples was a lot for us to handle. We realized that, this was something that you had to be very careful with, because as we were pushing this data through, little mistakes can end up costing a lot of money on the cloud. And I'll get into that, I guess, in a little bit. Next slide, please. So yeah, so this large amount of samples, we really had to shift to Google Cloud. So we started, moving our HPC kind of pipeline directly into, Fire Cloud, which became Terra, the Terra platform from the Broad. And so, that has its own challenges, but, we were able to do that, and it wasn't too crazy. And then we were actually able to process and run all 15,000 samples with this. And, and from that, we were able to get these, nice distributions to kind of match what we expected and what we had kind of seen in our previous HPC studies. And so we were kind of happy. We're kind of happy with that kind of transition from HPC to the cloud. Okay. So really, I just want to get at kind of what I've learned from using the cloud for genomics. So some of the benefits I found is that, the data sharing has become so much easier with the ability to store files on the cloud. You can really just point people to a link, get them access to a folder, rather than having to spend months trying to download things on Globius or something. It's just really much easier to share data with the cloud. We also have this, amazing ability to parallelize. And so, that's really is one of the reasons why for these large scale data sets, using the cloud is so important, is that you can't run a lot of these things on just a local computing cluster anymore. You really need the resources available to the cloud. I also really like the reproducibility of the code. So, we've always had issues with environments, and I feel like things like Docker and stuff like that have really made it a lot easier to kind of share code with other groups, and for everyone to kind of be able to run a specific pipeline, very easily without much work. I'm trying to, set up everything and trying to get their environment perfect to run any given program. And also, I think that this may be biased because we're kind of in the early days, but I've really found that the technical support for, whatever system I've been using has been spectacular. So whenever there's been an issue, people are more than happy to jump in. I think that there's just so many developers working on this that it's been quite amazing. So this is kind of stuff that from running a lot of different samples on the cloud, what kind of scares me. The first thing is really just the cost tracking and the lag behind it. We usually don't get reports for about 24 hours. So you run something and then you're just, you're hoping everything worked, and you don't really know exactly how much something costs until 24 hours later, which is a little scary. I mean, you can kind of estimate it, but you're still not 100% sure for 24 hours. And the other thing that scares me is that, to take full advantage of the cloud and the possibility of caching. So when something dies and, you want to use intermediate files in between, you have to store all those files. And, we've had issues where, just remembering to delete this intermediate data can, be a huge issue. And it's not necessarily always intuitive how to get rid of that data. The other thing is that, we've had a few issues where there's just been changes, like on the Google side, or, on whichever program or whichever software we're using, there's some sort of change that breaks the code, and you don't realize it until after you've run something that's kind of expensive, and then you have to go figure out what happened and, what's going on, and, is it possible that, you can get reimbursed for some of those on the cloud or whatever. And then, another issue we've had, and this is kind of out of, it's out of our control as users, but, getting surprise preemptible bills where just, you're running a intensive set of commands, on the cloud using a VM, but you don't really know that they're dying until kind of after you look, and you can see that, you're running on these preemptible nodes to try to save money, but the preemptible nodes are very busy at that time. And so you keep getting kicked off. You have to keep redoing these jobs over and over. And then you find this massive bill at the end of the day that, kind of messes with your benchmarking completely, because, you expect a certain amount when you're testing. And then you find out that, just at the time you happen to run a command, the cloud was very busy at that time, and that really caused issues. And then, you know, the other thing that always scares me is just scalability issues, because as we run larger and larger cohorts, you know, every little mistake is amplified and the cost is amplified. And so, running how to run jobs at once, how many samples can you process at one time? These are all things that are very scary for me. Just because, you know, I understand the consequences of making a mistake are much different than when you're working on a high performance computing cluster, and really just, you know, the cost monitoring for all of this. So somebody's asked, what do we use as a testing environment? So typically we run like much smaller cohorts. So we typically do things in batches to help control for cost. And then within those batches, you know, we'll run like one batch and we'll benchmark and make sure everything looks good. We'll do costs, and then we'll do like a scale up 1 to 10 to, you know, 50 to however many we need to get across, just to make sure that, you know, we're not going to get like crushed by trying to run everything at once, which is almost never a good idea when you have a lot of samples. So these are just some of the general challenges that I've had with interoperability. So so one of the things is that you know, we've written GTK-SV very specifically for the Terra system, and you know, the problem with this is that it can't directly access the data on AWS without pulling down to Google Cloud. And so, you know, we're kind of in the situation where, you know, do we pull these files down quickly to Google Cloud, run our stuff quick, and then delete them? Do we try to, you know, set up part of our pipeline on AWS and try to get the stuff, you know, that stuff run, and then pull it down to Google? Do we try to set up the whole thing on AWS? If we set up the whole pipeline on AWS, are we responsible for the support for both the AWS version and the GCP version, or do we just have to do one? These are the types of things that we're having a challenge with as we're dealing with, you know, these really diverse different data sets, different kind of platforms. And then, you know, we spend a lot of time optimizing everything we do. And that's really, you know, it's time consuming, but it's really important. And, you know, if we are working on two systems, do we have to do that for both of them? And I think you do. And I just don't know if we have the ability to handle that. Okay, looks like there was a question. The first benefit you listed for using the cloud was the ease of sharing data. How were you granted access to others via GCP or AWS? Data mechanisms and storage you manage, or some other approach? So my comment about sharing data was kind of like, it wasn't necessarily from any of these platforms. It was just, you know, like internal data we have, or you know, you can really just give access, you know, for a specific bucket or not, that you want people, you know, have access to, and then you can switch that access once they actually, you know, download data to whatever they're working on. Or if they're both working on the cloud, you can just give them access to your BAMs as a reader. So there's just a lot of flexibility with having the storage, and it's, you know, it's very easy to move things within Google Cloud buckets or AWS buckets, but it's not very easy, you know, before, when you have to like go across systems. Okay. Okay, so I guess I'll just wrap it up. So I've helped build a cloud-based SV pipeline that's been applied to tens of thousands of samples. You know, these studies are really only possible in the cloud. And I think that, you know, the cloud has really transformed what we can do, and it's quite amazing. So I think that, you know, and it all starts with, you know, the developers and you guys that are working all this work out, because, you know, I'm really coming at the back end. I know all the hard work you guys have been doing to, you know, think of all the questions that I don't know. And so it's been quite amazing. And, you know, the one thing I just want to point out is that I'm still terrified of the cost of using cloud. Always, like, it's just, it's a scary thing.
To that end, I just added a question.
Okay. The G4GH RAS passport mechanism could be extended to carry various permissions. Should we be looking at controlling costs? So I guess I'm not familiar with that. Is that for sharing data, or what specifically is the passport?
So GA4GH passports with visas, you know, what they do is they establish essentially who you are and what data sets you should have access to.
So Brian, I can answer this. A GH passport is a cryptographic token that handles the authentication part of identifying who you are, as Brian was saying. And then it carries a set of permissions where these permissions are arbitrary. They're permissions to do anything that you can describe, and they can come from different places. We focused mostly on having permissions to access data, but the act of incurring a cost or generating a bill is something that itself could require permission. And there are ways that this could be integrated into the system going forward that might make it more difficult to run up these costs that you're talking about.
I think it's a really great question. We actually have a session on Monday specifically around this. And then I'll actually be introducing some of the interactive stuff we're doing this afternoon where we can maybe capture some of these things. I do want to kind of push on so we don't run into lunch too much. If you had any last things, Harrison, otherwise we can move on.
Nope, I'm great. Thank you.
Questions in chat, and feel free to continue having the discussions in chat, and we will move on to Elisa Manning and the work that she's been doing.
Hi there, everyone. Thanks for inviting me to come and present. I think there was a hope that I would be able to attend a previous NCPI conference, but wasn't able to make it happen. So, here we are, and I'm really excited to be here and learn from all of you. So we're going to be presenting today a proof of concept for interoperability approaches for improving outcomes of pediatric diseases. So this is a project that we're doing at the Broad Institute. Tim Majarian is going to be doing the majority of our presentation, but we're working with Brian O'Connor at the Broad and other developers to develop the steps that we would want to implement. And then with many of you guys, we're working with you to get those go through these steps from a researcher perspective, not trying to use, basically trying to be true to the experience that people would have on the platform coming as a researcher. So if we go to the next slide, I'll give just a really brief kind of overview and background of where we have been using the cloud for complex trait genetic analysis. It really started in 2017 when the TopMed whole genome sequence data became available, and we as researchers in the diabetes space became ready to do analysis. So it took us some time to get our traits in order, and when we were ready to kick off analyses, we wanted to do genome-wide association studies and rare variant association studies with the whole genome sequence data. So we wrote our first WIDLs and deployed our first cloud-based workflows on the Fire Cloud system, which hosted at the Broad, was one of the TopMed cloud computing pilots, kind of early days of us putting our feet into the water, so to speak. So then in 2018 to 2019, we really got going doing collaborative research in the cloud and got used to some of the mechanisms and ways of sharing data with our collaborators while staying within consortium and data use rules. It's been one of the major successes of TopMed whole genome sequence data analysis that we've been able to figure out ways to do collaborative analysis, and it's just been really nice being able to dive into a workspace and help debug one of your collaborators who you're working closely with. So in that time period, we really got rare variant analysis workflows up and running. We collaborated with folks at the Analysis Commons hosted by DNA Nexus to get workflows written in WIDL, registered in DocStore. I started a project developing methods for large-scale gene-environment interaction, and we started off in our project proposal planning to distribute our software and workflows in a fair manner. So from the get-go, we were putting things on GitHub, developing WIDLs and workflows deploying them to the cloud platforms and running our analysis there. And we also participated in some major initiatives at the Broad Institute and the Terra platform to educate people about how to do genome-wide association studies in the cloud. And we have developed a featured workspace that's available in Terra. And Tim ran a workshop at last year's American Society of Human Genetics, which was well received and I think really fun for us to do. So now here in 2020, we're working within Biodata Catalyst, where I'm a new principal investigator on the project from the Broad Institute. I have a postdoc who is a member of the Fellows Cohort 1, and in fact, I collaborate with Harrison as well. So there's lots of interaction in my group with folks who are in the like the early stages of using Biodata Catalyst. And today we're going to be presenting this CSCI Interoperability project, which is really a pilot to process, we're piloting the process for doing cross-platform analysis. With that, very quick background, we're going to turn over to Tim, and Tim is going to present, and I am going to maybe monitor the chat a bit if people want to have, if you have additional questions.
All right, great. Thanks, Elisa. So I'm Tim. I'm part of Elisa's lab at the Broad Institute, and I'm really going to give a little bit of a background and kind of a status update on this initial project. So like Elisa said, the kind of idea behind this is to demonstrate how a researcher from our field, so human genetics, would come in and try and run some sort of analysis on data that exists on different cloud platforms. The specific aims of the study were really to identify, access, and kind of take a look at all the data that's available that we've kind of picked out to run this analysis. That's what you see in the table on the right. Listed are the different data sets that we use in this project. So between Anvil, the Kids First Data Resource, and Biodata Catalyst. The next kind of aim of the study is really to leverage the individual level data that's available on different cloud platforms to assess the contribution of rare variants towards congenital heart defect risk. So it's a pretty basic study. We don't have huge sample sizes, so it's really just to demonstrate how a researcher would utilize these different types of data to really run an analysis. The framework that we're going to utilize is often referred to as, say, using internal cases and external controls. This is kind of a process that's pretty common within human genetics, especially when we have these small disease-focused studies where, say, sequencing is only done on people with a disease or phenotype of interest, and we might want to run an analysis that also uses, say, controls, people without that phenotype. And so here, the internal cases will be from the Kids First Data Resource, and external controls will be from the Top Med study, Framingham Heart Study, and Jackson Heart Study. I mentioned here also that we're going to use GTEx data. That won't be in this presentation. That's kind of a future thing that's written into our analysis plan. The method that we actually use in this project is called ProxyCat. It's designed specifically for using these external controls for association testing. And the basic method is to compare, say, the number of rare, synonymous, and non-synonymous variants within genes between your case and control cohorts. So next slide. And so really the first step in this project is to export all the data that we need from the individual platforms into their native cloud compute areas. So here's just an example of how we get data from Biodata Catalyst. You can go to the next slide. Here's the same thing for Kids First, where we export to Kivatica to do the initial analysis, and then next slide. And the same thing with, say, data from the Anvil project for GTEx. Okay, next slide. And so the first step in any one of these analyses is really to get to know your data. So for this, we utilized each one of the individual cloud platforms. So on the left, you can see for Biodata Catalyst. In the middle, we have the Kids First data on Kivatica. And then on the right, Anvil, the GTEx data that we use through Terra. We can run basically the same type of analysis on each one of these platforms using their interactive kind of notebook support that they have. And here's just an example of something that we might output from that initial phase of really getting to know our data and understanding what's there. So here we're just showing age versus reported race or ancestry, and then subset by, say, gender or sex. So next slide. So the next step in this analysis is really to prepare the genetic data for the actual association analysis. So again, here, just like those phenotypic summaries that I showed, all of this preparation is done within each individual ecosystem. So we work with the genetic data, say for Kids First on Cavatica, and then we work with the genetic data that comes out of Biodata Catalyst on Terra. So to subset down to the variants that we actually want to look at for these association analyses, I said before that we'd look at rare variants. So this will be variants with minor allele frequency less than 1%. The Proxycat software also requires that we have kind of two types of variation. So functional and non-functional, or synonymous or non-synonymous. So for this analysis, we'll only be looking at variants that are within, say, protein coding exons. So then the next step would be to actually annotate these variants. So to figure out which variants are synonymous, which ones are non-synonymous, and what genes those kind of correspond to. For the Kids First data, we use a specific field from the VCF files that are delivered to us. This is the annotation field. Basically just has predictions for variants effect on gene expression, protein function, that kind of thing. For the TopMed data for Jackson Heart Study and Framingham Heart Study, we had to bring in an external resource. So this is this DBNSFP database. It basically has the same predictions that you would have in the annotation field for Kids First, except within the TopMed data, that annotation field isn't actually there. And so lastly, we next need to count the number of variants that are within each gene within your genome, right? So the actual association test is to look at the differences between the number of synonymous and non-synonymous variants between cases and controls. Okay, so next slide. So here, after actually running this Proxycat method, we get some results. Some important things to note at the beginning, though, is that we ran this association analysis only within Biodata Catalyst. So we actually brought the data from Kids First and put it into Biodata Catalyst, so into a Terra workspace. I'll touch a little bit more on that on the next slide. But as far as the results, we tested over 17,000 genes for association with CHD risk. 55 of those passed our Bonferroni corrected significance threshold, and one of those was a known CHD gene. On the right, you can see the Manhattan plot. So this shows p-values for each gene, as well as their location along each chromosome. In green, these are the known CHD genes. And then that red line across is actually the significance level. So you can see one of those dots that's above that significance level is actually green. So that's the one known CHD gene. And so this was just kind of a first pass analysis. So there's obviously some more things that we could do, specifically like, dividing out by ancestry or race. Right here, we're just using a full pooled sample. Okay, so next slide. So after kind of looking at these results, I thought it would be helpful to kind of look back at, how we might have done this analysis prior to interoperability features, how we did it right now, and what things might be useful in the future. So before, say, any of these interoperability features, we had a regular paradigm for running this type of analysis, and I've kind of divided into a few different categories. So first is data authorization. So obviously we have to go to dbGaP, apply for access, and actually gain that access. And then when we actually want to grab that data, we need to go into dbGaP again and create some sort of download request. How we used to actually get the data to a cloud platform. So when we're using Terra or something like that, we would reserve a Google Cloud VM, use a Sparrow to download that data from dbGaP, and then subsequently upload that data from our Google Cloud VM to a workspace bucket that is accessible through Terra. And so that's obviously, it costs a bit to do that. It takes a lot of time. It's pretty cumbersome. And then how we would actually do the data pre-processing and final analysis, we do it in a single Terra workspace. So that was kind of before we had any of these interoperability features that we were aware of. And so here's how we kind of did it now. So getting authorization is similar, right? So to apply through dbGaP and actually get that access, except now really getting that data to the cloud platform that you want to work on is just so much easier. So a single login using your ERA credentials rather than say, starting a VM, downloading it or using a Sparrow to download the data and then uploading it to a bucket. Here we can really just export those links directly from those data sharing platforms, right? Gen 3 for either Anvil or BDC, and then for Kids First, that Kids First data resource. And so again, that's really much easier than the kind of paradigm that we were working on before. And so for these last analyses that I presented, I said that we used separate workspaces within these individual ecosystems to do the pre-processing. So say we pulled out all the variants that we wanted from the Kids First data on the Kivatica platform. Same thing for Biodata Catalyst, except with Intera. And then for final analyses, we used a single Biodata Catalyst workspace that kind of linked to the other ones that we were using. And so here is where that kind of downloading the Kids First data came into play, right? So we still didn't have a method of accessing Kids First data from within the Biodata Catalyst ecosystem. So we had to kind of rely on an older paradigm, right? So specifically downloading it to my local computer and then uploading data back up to the cloud so we could actually use it in some sort of combined analysis. Obviously that's not ideal, but yeah. So then in the future, things that we think would be useful and would streamline this process of running this sort of analysis. Obviously the dbGaP stuff doesn't change, still have to gain access. But really having a single sign-in within, say, one ecosystem would be very helpful, right? So rather than jumping between, Kids First and Kivatica and then moving over to Biodata Catalyst and Terra, it would be really easy, say, if we could just have one sign-in and be able to access all of that data from a single portal. The data pre-processing as well would be a little bit less cumbersome if we could do it all, say, in the same workspace. There's some issues now with, say, the way the data is exported into a Terra workspace. They don't always line up, say, between Anvil data and between Biodata Catalyst data. And then for the final analysis, again, we'd want to use a single workspace just to keep it easy and remove this kind of like downloading data locally and then uploading it back up, right? Ideally, there would be a feature where we could access data that exists, say, through the Kids First stuff directly from the Biodata Catalyst ecosystem. Okay, next slide. Okay, and then lastly, I just thought it would be, helpful to talk about some of the stumbles and maybe the roadblocks that we ran into in the initial phases of this analysis. So a few of these are solved, some are still, outstanding or, might not be fixed, but these are just things that we've run into. First, data availability across platforms. So I talked about this kind of accessing that Kids First data from within the Biodata Catalyst ecosystem. That would really make it easier for us, not having to do that download and upload. A few that have been solved. So say we had some issues with PFB import into Terra for the TopMed PCGC data. Thankfully, this has been solved by some awesome work from people here. Fixing these DRS links from GTEx. So that's also been solved, so we can access that data easily now. And so two other things that I've kind of noticed that, still exist. So workflow compatibility. For example, on Kivatica, workflows are run in CWL, whereas on Terra, we run things in WDL. Ideally, we would be able to, run the same workflow in both places, just so we know that, we're getting the same results. Or say we have two different data sets and we want to run the exact same pipeline on those two data sets. It would be nice to have a way to actually do that within both ecosystems. And then finally, data documentation. So all of these interoperability features have really made it just so much easier to actually gain access to this data and to really touch it and work with it. It's been totally awesome. But one of the things that kind of remains a little bit difficult is actually finding very particular information about these different data sets. So I put some examples of, just questions that I've kind of run into during this project. So one is, why is that annotation field missing from the TopMed VCFs, but it exists within the Kids First data? Kind of finding an answer to that is a little bit difficult. What fields are actually available within these VCFs or these genetic data files that we're using, and what do they correspond to? That's something that you can go and look up, but it would be easy if it was readily available. And then a final one, just what methods were used for genotype calling, say between the Kids First data and the TopMed data. This is something that is particularly relevant to running a genome-wide association study. We would want to know how variants were called. And okay, I think that's it. And just thank you to everybody that helped so much, particularly Brian, Michael Bauman, and Allison, who helped us a lot with the Kids First data. Yeah, so if you have any questions, I'd be happy to answer.
We have time for one or two quick questions, if there are any, either on the chat or voice.
Tim, I've got a question regarding how those study-specific questions are hard to find. In your dream world, as a researcher, how would you find them? I mean, I think right now I'd say go ask Albert at the TopMed IRC, and he could give you those details. But what would be a good way to do that?
Yeah, so I mean, I think standardized documentation for particular file types or data types, right? Like there are certain questions that you might always want to know with variant calls, right? And it would be, what is the sequencing depth on average or something, or, what program was used to call these variants? So I mean, I think standardization is basically the answer to that question. Yeah.
Great, thanks. There's no other pressing questions. Feel free to continue it in chat, and we'll move on to actually learn even more about congenital heart disease from Daniel Crate.
Hey, thanks to the organizers for inviting me today to talk about my experience using the cloud computing platforms. So I'm a pediatric cardiologist at Boston Children's Hospital and a postdoctoral fellow in the Seidman Lab at Harvard Medical School. And today I'm going to talk to you about using cloud computing resources to study structural variation in congenital heart disease. So a little bit of a combination of the first two talks. So first I'll introduce the clinical question we're trying to address, and then talk to you about my experience using these platforms to try to figure some things out. So congenital heart disease is the most common congenital anomaly and the leading cause of mortality due to a birth defect. As you can see, this GIF of a heart on the right. Normally the heart has basically septa in the top and bottom chambers that divide it into the left and right side. And as you can see in this video, this heart is missing parts of those walls. It requires surgery in infancy for the child to do well. And there's a strong genetic basis for congenital heart disease in that many genetic syndromes are associated with congenital heart disease, and congenital heart disease is frequently seen in many chromosomal abnormalities. Next slide, please. And so over the past decade and more, a lot of effort has been put into trying to understand the genetic basis of congenital heart disease. And this slide summarizes our current knowledge of the genetic causes of CHD. So many cases are caused by aneuploidy, so gains or losses of whole chromosomes. Many cases are caused by CNVs, or copy number variants, which are duplications or deletions of large stretches of genomic DNA. A smaller subset are caused by very small single nucleotide variants. Some are environmental due to sort of maternal medication use and other environmental factors, viral infections. But as you'll notice on this pie chart, the majority of cases remain of unknown etiology, despite there being a strong evidence that there's genetic contribution in many of those cases. Next slide, please. And so our aim is to use whole genome sequencing to try to identify previously undetected genetic variants that are responsible for congenital heart disease. And the Seidman Lab is part of the Pediatric Cardiac Genomics Consortium, which is an NHLBI-funded multi-institutional consortium working to understand the genetic basis of CHD. Next slide, please. And sort of as Harrison touched on in his talk, recent data has started to uncover genomic structural variants as a cause of both structural abnormalities in disease, and also as an important class of normal variants that are present in the healthy population. And so many of the very, very large structural variants that encompass entire chromosomes or large pieces of chromosomes have long been studied in congenital heart disease. But if you can see this figure on the left, many of the smaller variants in the purple region have gone undetected based on previous testing methodologies. And so we were interested in studying structural variants that range in 50 base pairs and larger using whole genome sequencing data. Next slide, please. And again, as Harrison touched on, detection of genomic structural variants by whole genome sequencing is a resource-intensive process. It requires collecting multiple different levels of evidence, many of which are shown here, across the entire genome for many samples. And it was this resource-intensive nature of calling structural variants that pushed our lab to look towards cloud computing to try to address this question. Next slide, please. And so just briefly, I think our lab has long used a high performance computing cluster for sort of all of our genetic and genomic analyses. And this was our first consideration to use in the cloud. And I think some important points that eased that transition for our lab. So we had concerns about cost. We really had no idea what sort of cost was going to be associated with these analyses. And when we used the local cluster at our university, compute time is essentially included in overheads, that it's really at no additional cost. So having funds available for pilot studies was really crucial in helping us explore the option of doing these analyses in the cloud. The second thing was there was a learning curve. Nobody in our group had done cloud computing, and it's definitely a slightly different ecosystem, and there are different considerations. And so having user-friendly tool editors on Kavatica platform and the Seven Bridges team, bioinformatics team being available and really helpful to help troubleshoot was really a huge help in this transition. And then the last point is just data availability. The data was in the ecosystem, it was easy to access, and that made the ease of transitioning into the Kavatica system much less painful. Next slide, please. And so for our analyses, we often use trios. So we, for the PCGC, we recruit parents and a child. So the child has congenital heart disease, the parents usually do not. And so we had 716 congenital heart disease trios that were sequenced as a part of the Gabriella Miller Kids First program. And then for controls, we used what we call trios from the Safari Autism Consortium. So Safari has recruited quads, so an affected child, an unaffected sibling, and the parents. And we basically use the unaffected sibling and the parents as non-CHD trios in our study. Next slide, please. And so in order to do these analyses initially, the plan was to use the cloud ecosystem to do the heavy lifting and the heavy computational tasks. So for our CHD cases that were in Kavatica, we ran a list of structural variant calling algorithms, some of which are listed there. And on our local cluster where our Safari non-CHD controls lived, we ran sort of identical workflows there and did a lot of sort of comparison between the results that we were getting on our cluster and the results we were getting on Kavatica to make sure that everything was compatible. And then we took the output of the tools from the cloud and downloaded those to our local cluster. And that's where we performed sort of the merging of the evidence supporting structural variant calls. We did additional joint genotyping of trios and filtering of the data set. Next slide, please. And in this first analysis, we really got some interesting results. We identified structural variants affecting, causing loss of function of genes that we know cause congenital heart disease. We found de novo loss of function variants, so variants that appeared in the child but not the parents, and genes that are generally not found to have damaging mutations in the population as a whole. And just as an example, in patients with a subtype of congenital heart disease called tetralogy of Fallot, we found a handful of variants that were within this size range that aren't well detected by other methodologies that were likely the cause of their disease. And so these were really promising results. And as we were working on these analyses, next slide, please. Our whole genome sequencing cohort was continuing to grow in size. And so we started off with 716 trios sequenced by Gabriella Miller. That number grew in size to 892 trios. And at the same time, a large cohort was sequenced by the TopMed program, so over 1,000 other CHD trios. In addition, the work by Harrison and the Talkowski group to look at structural variants on a population level was being published. And so based on this expansion in the data set and the availability of now population level reference data for structural variants, we decided to alter our approach. Next slide, please. And so in collaboration with Harrison and the Talkowski group, we've shifted towards using the GATK-SV pipeline to genotype structural variants in about 1,950 congenital heart disease trios. And this is being done in the Terra platform. And I think, the GMKF data was manually uploaded to the Terra platform in order to run this pipeline. Next slide, please. And so to again, get at the importance of interoperability, for us, for our data set is really split between these two ecosystems, with half the data living in the Kids First ecosystem and the other half living a TopMed ecosystem. And so this is obviously a major barrier for running these large analyses, but also, the downstream work after you generate variant calls and things like that, and you want to go back to the alignment files and just take a quick look in IGV and look at the sequence alignments. You have to decide which platform you're going to go to depending on where those CRAM and alignment files live. So while it's a problem sort of for the upstream analysis, it's also makes the downstream work a little more painful as well. In addition to congenital heart disease, we also have our lab also has other data sets that straddle these ecosystems. So in the TopMed program, we've sequenced about 1,000 individuals with cardiomyopathy with whole genome sequencing. And then in the GMKF ecosystem, we have a project on microtia, which is a problem with ear development. And again, we're interested in applying GATK-SV, or we have applied GATK-SV to these data sets as well. But in order to do so, I've had to sort of download and upload files in order to get them to the correct system. And then, the other thing I want to touch on is that, this work has sort of been our lab's first introduction to cloud computing. And now as we look towards performing more analyses in the cloud, the location of data sets and workflows is really a major consideration of how and which ecosystem we spend our time developing workflows in. The next slide, please. So just to touch on, I think the big positives of experiences that I've had using both Cavatica and Terra platforms is really the opportunity to accelerate research through use of on-demand compute resources and really accelerate what would have taken me months to do on the cluster at Harvard Medical School to doing in a matter of weeks on the cloud. And then also, I think Harrison touched on this, just the ease of data sharing. As our cohorts grow in size, we're always looking for more non-congenital heart disease data sets to run for controls and as comparison. And as more whole genome sequencing data becomes available on these platforms, performing the analysis in these ecosystems makes accessing more control data much more feasible. And then just to touch on some of the barriers, as somebody who came into, this is my first foray into cloud computing where these analyses, again, the barrier of the data sets living in different places, to touch on Harrison's point of difficulty estimating cost upfront. So you do a pilot analysis and try to understand what your costs are going to be, but making mistakes in your workflows is expensive, and how do you budget for that? And then also the difficulty in monitoring costs. I think, as Harrison touched on, there's a delay in the bill you get. Also, I've run into issues with both ecosystems where there are back-end errors that leave compute instances running. And so potentially having things running for days that you really aren't aware of if you're not on top of your billing can the bill wrapping up. And then the other thing that was touched on by Tim, this sort of WDL versus CWL difference between pipelines. If you're going to invest your time, importing your pipelines onto the cloud, do you need to do it in both ecosystems, or do you choose one versus the other? And do you choose it based on where your data is? And so it'd be nice to have interoperability, both in terms of where the data lives and portability of pipelines that you generate. Next slide. And that's it. And with that, I'd like to thank everybody in the Seidman Lab, at Boston Children's Hospital, who have made the PCGC work possible, the Tolkowski and Brand Labs for their collaboration now on the GATK-SV work, and then Mark Walker from the GATK-SV team, who was very helpful in setting up Terra workspaces and providing little scripts to get that up and running.
Great. I think we've got time for maybe one or two questions for Dan.
So I think it's interesting to hear the kind of theme of data sharing is both easier, but data sets are still split between different places. And I actually am curious, I think maybe about like the data sharing being easier part. You know, is it easier to find the data sets, and even though you have to move them around, it's still a little bit easier? Or kind of what's your take on that?
Yeah, good question. I think we're in a little bit of a different situation because we still download everything to our cluster, and so we can also, upload is essentially free, and so you can upload, compute, and delete data, and so that's a strategy we've used. But as we're definitely looking at other TopMed generated control data sets, and so yeah, all you have to do is get your dbGaP access, and then you can pull those into workflow. And instead of spending your money downloading, thousands of cram files, you just spend the money processing them in the cloud. It's faster, and you pull down, the output at the ends to do your downstream analysis. So I think the data sharing and availability of controlled data sets, and then as our CHD cohort grows and the feasibility of literally downloading and moving data becomes much more of an issue, I think just computing on it in the cloud and having access to it wherever it may lie to get it into your workflows is going to be the way we need to go.
I think it's great, and I wonder whether what both you and Harrison were really suggesting is that because all of your analyses actually use data in addition to that data that's being provisioned or provided by the platforms, that the capacity to bring in that data and or share it even with collaborators within the context of the data provided by either Kids First or BD Catalyst, that process is actually also empowered, right? And that's actually a recurrent theme, is that I think it sounds like that most groups, in order to do their analysis, want access to the data being provisioned plus other data that they potentially have already curated, have at hand, have used as controls, and that you have to solve both those problems at the same time, right?
Yeah, and I think that was a big consideration in our initial analysis. We had the Safari cohort, which is a very large sequencing cohort, all on our local cluster, and so we could have said, oh, let's do all of this in the cloud, but we would have had to either get access to Safari in the cloud or upload it. And so it made sense to do some of the upstream heavy lifting in the cloud and then just do everything else where the controls lived, because for us, the control was the very large data set. But again, as both of these data sets start to grow and grow and grow, I think the use of the cluster for these analysis is becoming not feasible. And then the fact that the pipelines now just live in Terra cloud changes the dynamic.
Thanks. And yeah, feel free to continue the conversation in chat, and we'll move on to our last speaker and shift gears a little bit, but I think also to different cancer data sets, as well as some of the higher level analysis that's going on. So I'll turn over to Hoon Kim to present.
Hi, everyone. Can you hear me?
Yep, loud and clear.
Okay. Yeah, hi. But first of all, thank you for inviting me to the seminar that I have never joined before. And then the title of my presentation is Studies on X chromosomal DNA alterations using cloud computing of multiple tumor types. So I'm Hoon Kim, from Senior Research Scientist from the Jackson Lab in Connecticut. Actually, it's snowing outside. So two of my studies are made possible through the Cancer Genomics Cloud of the Institute for Systems Biology and ISGS data on Amazon Web Service. The first study, which is published 2018, involved the analysis of whole genome sequencing data from 53 TCGA glioblastoma and low-grade glioma samples. The second study, which is published recently, 2020, involved the analysis of whole genome sequencing data from more than 5,000 samples, including the tumor and normal. So here, actually, I'm going to briefly introduce these studies. The next slide. yeah, so the first thing is that I want to give very brief overview of X chromosome DNA, which is also called ecDNA. So ecDNA is circular DNA, which is different from chromosomal, the linear chromosomal DNA. So when you talk about chromosomal DNA, the chromosomal DNA is usually linear, but the ecDNA is circular DNA. And it is also referred to as the minute bodies or double minute, according to the study in 1965 through cytogenetic analysis of neuroblastoma cells. And previously, it was reported to be present in only 1.4% of tumors, according to Milton database 2007. And then that's why people were not much interested in ecDNA before. Next slide. So one of the biggest features characterizing ecDNA is that ecDNA is unevenly segregated in daughter cells during cell division. So let's look at the chromosomal alteration cases. So when chromosomal alterations cells replicate and divided into the cell, next slide. All the chromosomal alterations are equally divided into the cells. So in these cases, the parental cell have 3 copies of chromosomal alterations, and 3 copies are replicated, and then 6 copies, in total 6 copies are evenly distributed into the cell, with each cell carrying 3 copies of chromosomal alteration. Next slide. However, in case of external chromosomal alteration, the segregation pattern is highly different. So in these cases, the tumor cell carried 3 copies extra chromosomal DNA alterations. Next slide. When these tumor cells are replicated and segregated, all 6 copies can go to one daughter cell, and the other cell cannot carry any extra chromosomal DNA. Next slide. Or 5 copies of ecDNA alteration can go to one daughter cell, and the remaining 1 ecDNA alteration can go to the other cell. Next slide. Or 4 copies of ecDNA can go to one daughter cell, and the remaining 2 copies can go to the other cell. Next slide. Or like the chromosomal alteration segregation, ecDNA alteration can go to 2 daughter cells with equal segregation. Next slide. So because of this unequal segregation pattern, when tumor cells carrying X chromosome DNA, ecDNA, progress over time, at the time of tumor biopsy, the tumor tissue can get tumor cells with different extrachromosome alteration. So this suggests that ecDNA can drive intratumor heterogeneity. Next slide. So, but the important question when we try to analyze whole genome sequencing data through cloud is that, can you detect ecDNA from sequencing data? So answer is yes. And this is an example of ecDNA carrying the MYC and the oncogene. So if you look at the bottom panel, you can see circularization of the 3 segment carrying the MYC oncogene. Next slide. So we applied the computation approaches in 2 different studies to understand X chromosome DNA incidence as well as its evolution. So first study is to model GBM, which is glioblastoma, evolution in vitro and in vivo. So we have 13 GBM patients, and then for each GBM patient tumor, we generated cell line, and those cell line tumor cells were transplanted into 3 biological xenograft replicate. So total, we have 65 samples, including 13 patient GBM, 13 patient matching cell lines, and 36 xenograft. So basically, the original goal of this study is to examine whether genomic alterations observed in the patient sample would be preserved over the model system. And then, so this is a collaboration project with Anna from Heliport Hospital. We generated multi-type sequencing data from all the 65, but in particular, we applied the EC detection approach to whole genome sequencing data. Next slide. So we examined the presence of oncogene carrying ecDNAs over all 65 samples from 13 patients. So in this figure, y-axis represent the frequent, the GBM oncogenes. And then what we found out that all the oncogene amplification we detected from this data were actually X chromosome DNA. And then actually this is what was fascinating. And then also this suggests that ecDNAs might be highly frequent in glioblastoma. And also this study also suggests that the previous ecDNA incidence rate, which was 1.4% according to mutant database, might be incorrect. Next slide. And then we also analyzed additional 58 pairs of initial and recurrent gliomas to detect ecDNA and their effect on tumor evolution. And then the part of data set were accessed through ISV-CGC. So we had this data, so 27 pairs of glioma samples from TCGA, and then this cloud-based analysis was done when I joined the JAX, which is early 2017. But I had no experience in using cloud computing. But fortunately, our Jackson lab has a cloud specialist who is a Sandeep. So he gave tremendous help for analyzing these 27 pairs of glioma from TCGA through ISV-CGC. And then we realized, we again confirmed that ecDNAs are highly frequent, even the other, even the 58 pairs of initial and recurrent gliomas. Next slide. So next question is that how many ecDNA will present even multiple tumor types? So we analyzed more than 5,000 samples, including tumors and normal, which are available in TCGA or even the ICGC, the PCOT. So next slide. However, those data set was available on cloud. So challenges that we had to do large scale data analysis in hybrid and multi-cloud system. So initial and subsequent analysis was done in our local computer, but the analysis for those data set, the main analysis should be done in cloud computing. So we used the workflow engine such as the Chromel to avoid re-engineering the workflow. Next slide. So analysis of TCGA whole genome sequencing data was done on Google Cloud Platform. Basically, the clouding setup was done by Sandeep, our cloud specialist. Also, actually, we have tremendous help from ISV-CGC, Sheila, and other collaborators. So ISV-CGC hosted the TCG data in Google Cloud. And as I mentioned in the previous slide, we use Chromel workflow for this analysis. And in particular for this TCG data set, we used the Google preemptive virtual machine to save cost as much as possible, but still our cost was pretty high. Next slide. And then the ICGC whole genome sequence data was available in Amazon Web Service. And again, the Sandeep helped me to set up accessing this cloud. And we also used the similar Chromium workflow for analyzing whole genome sequencing data available on Amazon Web Cloud. But unlike the Google Cloud preemptive machine, the Amazon Spot instances did not have any such a limit of the analysis runtime. Next slide. Yeah, so through the cloud-in computing, we are able to analyze ecDNA of multiple tumor types. And then this is a collaboration project with UC San Diego and Stanford and the Boris Institute of Health. And especially I have a great help from the two postdoc and PhD students from the UCSD. Next slide. So the important finding is that the ecDNAs were present in 25 of 29 cancer types we analyzed, especially the high frequencies were observed in the most malignant cancer types, including glioblastoma, and sarcoma, and ovarian, and the breast cancers. However, ecDNA were almost no ecDNA was detected in normal, suggesting that ecDNA plays a critical role in cancer. And then also this study also confirms that the previous ecDNA incidence rate is wrong. Next slide. Yeah, because the ecDNA frequently carry oncogene, and then therefore the ecDNA carrying tumor samples have overall the worst outcome compared to tumor sample carrying oncogene, the oncogene chromosome amplification. And next slide. Yeah, so and then as of October 2020, the ecDNA was selected as one of the most important problem in cancer research by the Cancer Research Challenge initiated by the NIH, NCI, and Cancer Research UK. And then we hope that our lab can also join this challenge. Next slide. Yeah, this is a summary slide. So regarding our biological findings, the ecDNA contributed to intratumor heterogeneity, also present in multiple tumor types, and contributing to poor outcome for patients. In terms of cloud computing, this was very toughest part for me as a non-specialist in cloud computing. The significant engineering was required for setting up the resource on cloud providers, but fortunately we had a very wonderful cloud specialist. And then the workflow manager such as Chromel or Nextflow was helpful to avoid re-engineering the workflow. And next slide. So this is, I want to give special thanks to all patients. First, I really appreciate all the patients for providing the value of sample for research. And also special thanks to Roy Bach, who is my senior advisor, and also Sandeep. Also the Sheila and David and Bill from ISO CGC, they provide great help for me to access the cloud system. And thank you. And if you have any questions, please let me know.
Thanks so much, and thanks for joining us in the interop. I think once you're here, hopefully you can do useful things. Anybody have questions?
I wonder if any of the sort of birth defect or non-cancer groups have looked at extrachromosomal DNA.
Can you say again?
I wonder if extrachromosomal DNA elements have been reviewed in the non-cancer settings in any data sets.
Yeah, so actually, as I showed in the previous slide, ecDNA was not detected. Actually, only 0.5% of normal samples carried extrachromosomal DNA according to our criteria. This suggests that we barely detect ecDNA in non-cancer samples.
Yeah. I think in the non-cancer context, some of the groups are still looking at lesional sites that are non-cancerous. And that potentially is an area of interest.
Yes, actually that would be true as well, because our definition of extrachromosomal DNA, actually the size of ecDNA we want to identify is pretty long. So probably the shorter size of ecDNA or circular DNA could be present in the non-cancer cells.
Very cool.
I did have a question because you mentioned ICGC, and I'm interested in kind of your experiences using the ICGC data, because that is kind of non-NIH data, and kind of comparing and contrasting, getting access and leveraging that for your research.
Yeah, so regarding ICGC, actually, first of all, I'm sorry, actually, I don't know much about technical thing. For example, how we set up the accessing the ICGC through Picard, the Amazon. But what I want to tell you is that it looks like some of data is also available in Google Cloud, and the majority of data is available in Amazon Cloud. So we need to use a kind of hybrid system for this analysis. Then, the workflow manager based on the Chromel was successful to analyze data present to two different cloud systems, and then but all the technical setup was done by Sandeep from our Jackson lab. So I'm sorry I can't fully answer your question.
No worries. Even the downstream analysis, were you worried about batch effects or anything kind of across the TCGA and ICGC landscape with this? Or?
Oh, yeah, that's a good question. So yeah, so because basically the tumor types available in ICGC data is highly different from the TCGA data. I know the Picard data included the many TCGA data, but actually we excluded those TCGA data. We analyzed the data available in ICGC, and then overall we realized that the tumor types present in ICGC data has the less incidence rate of ecDNA. I think it's because the tumor types in ICGC data is less aggressive than tumor type present in TCGA.
All right. If there's no other questions, I really want to thank our morning speakers. I think this was a great way to kick off. And I always know early adoption has both its benefits of being able to be at the forefront and move forward, as well as being the first people to hit the pain points and the roadblocks. So I really think we all appreciate the work folks have been doing, and hope this pays off both for your research as well as how we can improve this in the future.
All right, well, I see I've got 1:30 on my clock, so I think we can go ahead and get started into our afternoon session. So thanks everybody for a great morning. And before we dive into our working group updates, I think we want to kind of restate some of the themes that we heard this morning, and then give a preview for what we're going to lead into in our discussion this afternoon. So I'm going to pass the microphone over to Adam and let him say a few words.
Yeah, and these are certainly opinionated on my side. So, again, if there are other thematic elements that are coming up, we'll continue to collate them throughout the day. But, what was really clear from the morning session was really, at least from my perspective, I'm sure others, that they're awesome, impactful, accelerated science is actually happening in the context of multi-platform cloud settings. And, I really thank the speakers for highlighting their experiences. it was clear that both quote unquote expert users as well as new users who have not yet engaged cloud are able to leverage the advantages of cloud platforms when supported appropriately and by documentation, as well as the platform's native support features. It's also clear that users still face what I think of as binaries in decision-making that still limit their full potential for harnessing platforms and cloud. These are sort of overarching themes that at least I heard in the talks. One key that came up multiple times that we'll have a separate session on is costs, and the way users have to navigate those costs, both from a position of not knowing costs, but also trying to decide between on-prem versus cloud and which cloud. And importantly, and this relates to the second point, where and from whom do I have my credits, and how do I support quote unquote other data sets? So what we heard multiple times is that decisions had to be made around where am I going to bring other data sets or other cohorts that I need to use in the context of analysis of data sets being provisioned across any of the cloud platforms, and how do I intersect them? So users actually had to solve two problems at the same time. One, how do I bring data sets across multiple platforms together? And at the same time, they also have to make decisions on when I do that, how do I intersect that with other data sets that may or may not currently reside in those platforms? And I think that's, again, an opportunity for us to help them navigate. And then the third, which came up across multiple talks, is the workflow language, whether CWL versus WDL, and where should I, one, invest in transforming my own pipelines, or are the right combinations of multiple pipelines available, particularly where some of the analysis don't yet have sort of community-based standards and people are leveraging multiple workflows. And really, I think what I heard the users ask is there a way not to be locked in by this? Is there a way not to be locked into a platform or an environment by the choice of WDL versus CWL? And really empower that interoperability across both workflows and data sets and platforms. Again, these are sort of my own opinionated context that's informed by the rest of the team on the DRC, but I think there's a lot of conversation happening in the chat we'll try and capture moving forward. But regardless, awesome job from the speakers, and really clear that you guys were able to leverage advantages provided by the cloud-based platforms.
Thanks, Adam. And then I think Allison was going to give us an introduction of how we're going to kind of continue our conversation this afternoon.
So as we go into these working group updates, I encourage you to check out this fun retro board, although really we're using it more as a fun perspective board of trying to capture some of the things that have come up. So we have 4 columns there. Hopefully it's pretty straightforward to use. We'll do a little bit more of a demo this afternoon, but I think as you're thinking of things, as things come up in the working group meetings, these are good places to put down data sets and initiatives that are driving interop, some of the 6-month things that folks might be doing, 12 months, and then any emerging concepts or unmet needs that we hear. So just wanted to highlight that, and that's what we'll be using this afternoon. We'll talk more about that, but we should go ahead and move on to the working group update.
And that is linked in the agenda as well. So if you want to jot notes there while we're listening to the talks this afternoon, you can find that in the agenda. Okay, great. So now we're going to move into our afternoon sessions, and these are our working group updates. And so we're going to start with the NIH coordination group, and that's led by Valentina and Ken.
Thank you. So first of all, let me start by thanking everyone for being here today, and also congratulate everybody for the great work that happened this past year. So I think, personally, I think I am very, very impressed by the progress that we made in such a short time and under quite difficult circumstances. So I want to thank everybody for all of this. So let me say that I give this presentation on behalf of the NIH Coordination Working Group, which is co-chaired by me and Ken. Next slide, please. This slide shows you the members of the working group, primarily, actually they're all NIH staff members, that represent the 4 different resources of the NCPI and also members of the Common Fund Data ecosystem. We meet approximately every 2 weeks. Most times we meet alone, or sometimes we also invite leaders of the other working groups, or also maybe some of the people that are helping us to organize the workshops and other events that impact all the other working groups. Next slide. So I guess like everybody else, like the other groups, we've been trying to identify our set of core responsibilities. I think these 3 represent pretty much all the various responsibilities and roles that we play for the NCPI. So we do serve as a governance body for the NCPI. We provide stewardship for the various working group activities. And also we serve a liaison with various NIH offices. And I will provide more detailed information about every single one of them. Next slide. So with respect to serving as a governance body for NCPI, for example, we ratified the set of the NCPI interoperability principles that were proposed by the Community Governance Working Group. I'm assuming that Bob and Stan will talk about it later. We also try to balance the NCPI goals and priorities versus the goals and priorities of the IC-specific platforms. As you can imagine, there is some tension there. There are obviously deliverables that need to be done by every single platform. And those sometimes conflict with some of the activities that involve the same people that also work in the NCPI project. So how to do that? It has been a challenge, and I'm sure you hear about it, especially from people like Brian and Jack in the next presentations. We address issues as they come up. For example, one of the latest ones is how to grant access to the developers of the NCPI groups and to each other resources, trying to streamline the data access processes, trying to make it as less painful as possible. And then finally, and this it's actually extremely important. It serves as a forum for us to talk to each other and share information, which frankly was not in place about 2 years ago. There were lots of peer-wise conversation, but it was not happening as a, information sharing was not happening as a group. Next slide. So in terms of stewardship, of course, we helped facilitate, we helped to launch 5 trans-NIH working group, including our, after the workshop last year, there were a couple of months where not much was happening. We helped stepped in, we helped push things forward. And then, starting pretty much in January, lots of things happened all of a sudden. So that was great. We've been quite busy in organizing several workshops. So basically after the first one in October, there was another one in April, and then there is this one, and I hope there is another one in 6 months from today. Fortunately, and also we hosted a Train Your Colleague workshop. This was done mostly for the members of our resources to become more familiar with everybody's resources. So that was also an extremely useful exercise. And the last 3 events, the one in March, April, and October, these are all virtual. Next. So last but not least, we serve as a liaison with the rest of the NIH. So first of all, we make sure that we align our efforts with the goals of the NIH Strategic Plan for Data Science. We've done this in many ways. For example, we have certainly facilitated collaboration with the NIH RAS project. We actually been working very closely with the RAS project, and also our platforms are serving in the phase 1 of the pilot, and we've been providing a lot of interesting feedback, useful feedback to the whole project, and we're still serving as a testing platform. Second, we have been making, we've been working with Susan in trying to create funding opportunities for our activities. And I think, all the resources and platforms represented here have received supplemental funding both for work with RAS as well as for NCPI activities. And also, and this is something that affects primarily NIH staff and not much you, we have been working with, we've been requesting and have been successful in obtaining a data scholar that basically this is an individual that is being hired from outside the NIH with expertise in data science that will help us with the programmatic activities of NCPI. She is still in the process of being hired. She's going to come from the FDA, and she will be able to join our working group probably in December. So with respect to interactions, a lot of the things that we're doing have an impact and a repercussion in terms of decisions to be made at the policy level. And so we're going to facilitate interactions and decision making with various groups across the NIH that deals with policy issues. And finally, again, we serve as a channel, communication channel with the rest of the NIH. Next. So this is a set of goals. These are not in any way set by priority, but these are just some that we could easily think about for next year. First of all, again, trying to be better in handling next year's priorities and milestone than what we've been in the past, and make sure that we have a common understanding about that. The implementation of the interoperability principles, I think we have been discussing them. I think we have a good sense of what they are. I think now the proof is in the pudding. Are we going to be able to deliver on those or not? Then again, we'll continue to host workshops and trying to get together to, again, information sharing and agree on different goals and priorities. The next one, offer training opportunities for outside investigators. I think this is extremely important. I think, you know, hopefully by the end of this fiscal year, sometimes in September, it will be nice to try to attract other users that will help us to test out the interoperability tools that we've been developing. I have been talking to Alison some time ago about when she thought it would be a good time to do this. And so maybe it would be nice to hear if truly we can try to make this happen towards the end of this fiscal year. So pursue additional funding support. Again, we're hoping to be able to work with ODSS to do that and continue collaboration for us. Improve visibility across the NIH and share best practices for platforms interoperability across the NIH. Again, this is a question that has come up earlier about right now with just our 4 platforms. We're working a little bit with a common fund data ecosystem, but we're hoping that what we're generating is going to have an impact across the NIH. So when is the good time to start to do that? When is it that we're going to feel comfortable that we have something to share? And finally, solidify collaboration with the GA4GH work streams. There's clearly a lot that we're leveraging from GA4GH. We're trying to utilize in our platforms. At the same time, there is a lot of knowledge sharing that we could do. So trying to have a closer collaboration, I think would be useful. This is basically my last slide. So I'd like to hear from maybe members of my group if I missed anything, they would like to add anything to what I said.
Thanks, Valentina. And so moving ahead then to our next group is the Community and Governance Group, and that's chaired by Bob Grossman and Stan Ahol. So I'll pass the mic over to them.
Thank you. Can we have the next slide, please? So we have a large group, and we'll give them time to add comments if they want at the end. I just want to, in the first slide, present a high-level overview of what we've done. As Valentina mentioned, the principles of interoperability were approved a couple meetings ago. What we focused on is how to make them precise enough to determine whether a particular platform was following them, um, and um, um, and we're doing that in several ways. We created some definitions and we created a series of questions, and we've tried to summarize in a table how they did that. Once we agree on the questions and the right way to categorize things, we're going to encourage the platforms to expose an API so that they can return programmatically, for example, very specific things like what's required and sort of to interoperate with them, which platforms they interoperate with, et cetera. We'll go into this in some detail, but what we did in this past multiple months was try to make these principles precise enough so that they could be evaluated platform by platform. Next slide. So I put here for reference the principles again. We focused on 3 of the principles, for example, one of the principles very broadly was if you take data from another person's platform, they in practice should be allowed, in principle should be allowed to have access to data in your platform, assuming that the security and compliance of the platforms are compatible and that there's presumably some kind of evaluation of the security and compliance by each platform of the other platform. And we, this and perhaps other conditions such as interconnection security agreement could be wrapped up into an agreement that 2 platforms could agree to between themselves that would allow the mutual sharing of data for a particular type of data in a particular type of environment with a particular type of security and compliance in place. That was called a trust relationship, and that's how the principles were drafted. But to take that to the next level is what we worked on. And so we have a white paper that's available. The short name for this is the Cloud Platform Interoperability. It's Characterizing Cloud Platform Interoperability, or C2PI. So the white paper has a recent name of Towards Characterizing Cloud Platform Interoperability. It's not finalized, it's a working document, but it's trying to take us toward these definitions and sort of evaluation through a series of questions that each platform could sort of self-attest to. So that is available through the link. Again, it's a work in process. Next slide. So we're not there. We have some blockers. I think any platform in the world is happy to take other people's data, but most platforms are not so happy to let other platforms have their data. And so, one way to look at this is, let's say that platform A has data A and platform B has a user who's authorized to look at data A. And it doesn't have to be copied, it could just be DRS identifiers. And let's say that one way or the other, the environment in platform B that the user uses is authorized to look at data of a certain sensitivity, then what's required between platform A and B for the user in platform B to touch the data? This is still open. We're still meeting lots of situations where this is difficult. Similarly, if there's a user in platform C who's authorized to look at data A and the environment is authorized, can the user in platform C reach to platform B where there's that data, or does it have to go back to the originating platform, platform A? The platforms have rules that require that even though data is available on platform B and platform B, the user has credentials there. The user might, in platform C, who's authorized to look at the data, might have to go to platform A. So that's a lot to digest. So we've broken these down to some questions. Next slide. So I've talked about trust, and in the last few months, we're taking steps to defining trust. There's something that was already present in dbGaP, which seems relevant in today's cloud platforms. In dbGaP, I'm going to simplify it, but there's a notion of an environment that's authorized by the user's IT director, who sort of authorizes an environment that's safe for that data to be downloaded into dbGaP. And then the signing official at the university signs off that the user is going to be using that data in that authorized environment. That's a dbGaP model, and it uses this notion of authorized environment. And we can ask the question, and the working group was discussing this, is there an analogous notion of authorized environment for cloud platform? So certainly within the boundaries of a cloud platform that are part of this conversation, that cloud platform itself provides an authorized environment for users' data. The question is that the only place in a cloud that the user can analyze that data, that is, can it never leave that environment? And for most of us on the call have been thinking about these kind of problems for a while. I think we naively had this idea that if that data would be in the cloud, in DRS, in secure and compliance environments, users would be in authorized environments, and if the user was in an authorized environment, they would have access to the data no matter where it is. So there's a lot of times when we write our security plans, we have we use this model: mark the data and trust the marks. That is, mark the data with how it should be handled, and the system should trust those marks and handle it. Here you could almost say authorize environments through a CISO or some other process, authorizes users through dbGaP or whatever process, and then the systems that should trust that the environments are authorized and the users are authorized to do the data and let them have access. Next slide. So, we looked at questions such as, what different, for each category of questions, it's relatively easy, and there's consensus, especially with RAS, of how to authorize users, but there's a lot to decide on what does it mean to authorize an environment, and we spent a lot of time talking about that. Next slide. So I'm not going to go through this, but this is an attempt, and this is, we did this fairly loosely the first time, but we're going to, in the next couple of weeks, we're going to do this table more precisely with a common set of questions. There'll be a question for each role. There's a question about what does it mean for a user to be authorized? What does it mean for an environment to be authorized? What are the rules for egressing data by download? What are the rules for egressing data by access to another cloud platform? What APIs? And we'll do this for each of the platforms, and this is a pretty big step forward because this makes clear what the policies are for each of these platforms for these important questions. Next slide. So here is sort of a picture, and I'm not going to go through all this, but let's say there's a workspace W, and that could be in platform A, could be in platform C, it could be in another platform. But let's say that that platform is authorized. Let's assume that the user is authorized to access data D from platform A and data E from platform C. Then you can ask, under what conditions can the user bring in the authorized workspace, bring that data together and make a discovery, put it in Nature, and win a prize? So, so there's various notions that the environment can be authorized. If that environment is in the in a user's home institution, then the IT director through the signing official would authorize it. There's nothing wrong with the IT director authorizing a cloud environment, in which case this would be permitted, even though that's not one of the core cloud platforms. That's B. The platform A, the institute or CISO associated with platform A and C could authorize the environment. You could argue that if the environment is in FISMA moderate with 853 and has a pen test and a 3-party assessment, then that's enough for the data to be moved there. Or you could argue FedRAMP, but these are the kinds of questions we're going to ask each of the platforms. Next slide. You can ask a number of other questions. Each of these scenarios I'm showing came up in the different platforms and block people's access to data. Here's, is an ISA required? Is a trust relation? Can you chain these things? I'm not going to go over this. We're out of time. Next slide. So in the roadmap, we're going to complete the CTPI paper towards characterizing cloud platform interoperability. We're going to formalize the questions, redo table A with the questions. We'll define an API so cloud platforms can self-attest how they answer these. And then we'll work towards a policy that allows authorized users in authorized environments to access particular types of data, and we'll encourage the platforms, the institutes to be very clear on which platforms are authorized. And then we'll work towards getting some of the NCPI platforms to trust each other. I'm going to turn it over to Stan, who's the co-chair.
So Bob, I think you've covered things very thoroughly. I just wanted to add just a brief comment that I think the committee worked very well and very diligently on trying to make progress, and Bob did a great job of keeping us on track. So I think we've made progress here, and we can see from the morning sessions where we still have lots of work to do, and some of them are going to intersect some of the issues, and I'm trying to add to the Fund Retro board. Some of the issues that we identified this morning are going to continue to be interacting with the issues of governance. No big surprise there.
I just want to respond to Jamie's question. Can we go back one slide? One more. So this question is still relevant and still a blocker for a lot of people if there is no platform C. And this is still, so Jamie raises the question, can data be combined? And obviously that's relevant and there are questions about this. But for those of you, especially in the last couple of weeks, a lot of us were surprised that it is still quite hard that if there's an author, that if a user is authorized to access data through dbGaP, and it's clear that if the data is controlled by platform A, that data can be examined in platform A. But even without data E, you could ask the question, is there any process in which, other than if it's done by the user's signing official in a cloud environment, that's not then they're generally available to others. Is there any process that user who's authorized for the data, that workspace could be authorized so that user can analyze data D for platform A? For example, that workspace may be in platform C. And, in special cases when the author of environments or in one of the NCPI platforms. I think we're out of our 15 minutes. Is that correct? Or if there's any more time, we can have a question, but I think we're out of time if we started at 12:45. I can't remember.
There's time for maybe one quick question. We did start a little bit late, but so I'll open the floor.
This is awesome work, Bob and Stan. Paul Aviak speaking. Is the purpose of the white paper to propose answers to those questions, or is it just to ask the questions? What did you have in mind?
No, it's a little different. It's to get questions precise enough that the platforms could give us their answers. And we hope by seeing that matrix will give us our interoperability. And then by making the questions precise enough, we've already seen that there's been progress and some platforms are beginning to rethink a bit. And in that rethinking, there is more interoperability.
All right. Well, I think we're gonna kind of stop the presentation here, but again, continue the conversation in the Zoom chat, and we'll have more time.
Sorry, I had a quick follow-up question, which is, do we imagine that these things would be policy-based or actual enforcement permissioning-based, or both?
I think both. Stan, did you want to talk? Was that you?
Yeah, I was going to say both. I think that ultimately, David, we got to find a way that we can enforce, enforce with little quotes in my fingers, actual adherence.
Yep. Okay. I agree. That is challenging and it's a long-term project.
Yeah. So that's why we're taking the intermittent step of having them self-attest through APIs so that, it's precise enough. If they say, this kind of environment is authorized and they say this is an authorized environment, and then there's some kind of policy that keeps them from doing that, that's a contradiction that we can use as a forcing function.
Because you're describing creating an API that will describe the state of what's happening, but today there's not yet the policies that would enable this.
Correct. So we're working on both.
This is Vivian, and I guess that I would push back a little bit that there are policies in place about the processes about data access. And I think it's just a larger conversation. So it's trying to figure out, because the policies are really based on people. And so it's really trying to make people within and their institutions accountable to what they agreed to. So there are sort of processes currently in place for some types of data.
And
thank you.
Yes, there's many policies for investigators accessing different kinds of data, but the level of those questions are the level of the platforms, which is very different.
Right, and I think that's sort of another sort of pinch point we probably need to talk through, because it seems that sometimes there's some crosstalk with that.
I think we're out of time. I really want to call out to the committee and to the co-chair Stan, because we've come a long way, and I think we're at a point that we can be precise enough shortly so that we can get a little closer to either using the correct policy, or if there's not a correct policy, using the appropriate policy. If there's not an appropriate policy, taking a step towards that. We may not need any more policies. We just may need a rule, basically may need to encourage the platforms to be clear of what their rules are for authorizing an environment. And that's not a policy, that's just asking the institutes and platforms what their rules are for authorizing an environment. We're out of time, I think.
Right, I think we are out of time. So thanks, thanks for that presentation. I think I'm going to move forward now to our next working group, which is the Systems Interoperation Working Group, and that's chaired by Brian O'Connor and Jack DiGiovanna. So I'll pass the mic over to them.
So thanks so much, David. Can everybody hear me?
Yep.
Okay. Love the pictures. Love the pictures.
Well, it has to be in character, right? Groot and Vin Diesel, it just fits.
Who is who?
Groot is my costume.
Between the two.
So we were formed. Ryan and I have given this talk a number of times to different groups. So we wanted to take out sort of the intro slides, and I think the beginning of the talk, the beginning of the session today was really great, showing the different needs for interoperability, from Adam and Allison's introduction with the pediatric cases, Harrison's work on SVs, Alyssa and Tim going through the PCGC data set, Daniel on congenital heart disease, and Hoon on the ecDNA. Very clearly, even from the first meeting at RENCI, there was a need for systems interoperation. There was a research driver there of, we would be able to do more as a community if we could solve some of the issues that Bob has just so elegantly laid out in the last session, and Valentina as well in the introduction. In the next slide, please. So we started from this idea, again, a year ago, of data portals could already connect to workspaces. So in the bottom right, there is the Kids First data portal, which was, this was late 2017, early 2018, is an excellent portal for faceted search, being able to understand a lot of the different Kids First data sets, form a cohort, and really seamlessly push that off to Kavatica. And there's other examples on this slide that were developed at different times, but right above it, if we go anti-clockwise, Gen 3 in Biodata Catalyst could push a cohort to Terra in Biodata Catalyst. The 3 different cloud resources in the CRDC were able to get information and search it in some different ways from the GDC. And then on the bottom left, we had put together an integration with the Proteomics Data Commons and the Integrated Canine Data Commons. So within one IC, it was possible to do this. And it clearly wasn't a technical challenge. We could build it, but the problem is to build the next interaction. So if we wanted to connect the Kids First data portal to the CGC, we would have to rebuild that entire flow. And then when we wanted to connect Biodata Catalyst Gen 3 to the CGC, we'd have to build it again, and it would be 3 different systems and all different arrows. So we wanted to look at in the next slide, as a working group, how can we spearhead technical improvements to really make this more interoperable so that we don't have N different ways to connect to N different portals, but also marry that with realistic researcher use cases. So we didn't just want to get into technical hypothetical discussions. We wanted to say, okay, we've seen these 4 researchers and 4 new researchers today and new researchers every day. What do they want to do? How do we enable it? And so balancing the technical and scientific, and also keeping everything rooted in what can we do in 6 months? Because we could also have a beer and a nice discussion of what would be possible in 4 years, what would be possible in 10 years. But really, as Adam showed in the beginning with the work with zero childhood cancer, people need this today, right? We don't need to think about what's in 6 years. We need these improvements to be coming through immediately. So there's links here in the slides if you want to see our charter or join in. Please do. We welcome more people. But if we can go to the next slide, please. We started with driver projects, and quickly we got to 8. And we saw one of them this morning with Alyssa and Tim's work, which is the one on the bottom there, spanning the 3 ICs. One that's been talked about a lot is spanning Biodata Catalyst and Kids First for PCGC. There's also multiple cases to Anvil for GTEx and other data sets. One of the things I want to leave some time until the end of our talk for is this idea of driver projects. It was great to get them. We definitely, Brian and I feel very strongly about keeping this researcher-centric, user-centric design, making sure we're building the things that enable the best use cases. But at the same time, we noticed over the course of this last year, some of these driver projects were able to move forward and had representation and had funding and different things. And other ones were blocked and they couldn't move forward. And that's perfectly okay. It's outside of their control. There was a policy issue, or there was one human responsible for 3 different use cases. So that human could only drive forward one. So one of the things we want to consider moving forward is getting to, these are our 2 to 3 driver projects. We'll keep a backlog of infinity other projects, that's fine. But just to set expectations of what do we think we're going to make progress on and what do we think we won't. And the next slide, please. So this was presented in April at the virtual meeting, 6 months ago. What were the challenges in tech that we ran into in this group? The first one was a standardized handoff mechanism. So this is not consistent with what I said about 6-month intervals, because I think we argued over this, and Brian can correct, and we argued over this for 8 or 9 months over what would be the right way to move forward, and very passionate arguments either way. But we did put the check mark after that. We made good progress on that in terms of the PFB and the light PFB for the current time period. Standardized data access methods. This one, I want to also give some room to breathe, because while DERS was available when the working group started, there were actually numerous improvements to DERS over the course of this working group, over the course of time since it was formed, that were improved here that made this really possible. And again, we're going to get into these later, but there were some concrete accomplishments there. As Valentina mentioned in her presentation, getting coordination between the work plans and technical timelines. I think we made some good progress on that between the working group and help from all the different ICs to help to continue to support this and get people's work plans aligned. And that really helped. We really saw a difference in the second half of the year compared to the first. And then there was some other progress in different areas. Some of them we couldn't make progress everywhere, but I think as RAS really made great progress that was more than we expected. We didn't get to a common metadata model. There's really exciting work going on there in multiple ICs, and for example, the CCDH is really driving that forward. But we did make enough progress on what are a few common values that would have to be included in any metadata model, that we could at least, the portals could agree on that and the workspaces could agree how to interpret those specific entities, if not an entire model. Next slide, please. So conceptually, what should happen, or what we wanted to build in the first year, is we have this user here in the middle. And if you click, please, she goes to search on one or more portals. So here she's going to search on the Gen 3 Biodata Catalyst and Anvil portals, and click, please. And those results can be brought back to a workspace, again, one or more workspaces. And if you click again, this is going to go check with the Gen 3 instances to if that user is authorized to get the data. And if she is, then if you click again, she can analyze the data and look at the results. So that's what we were hoping to get to with this working group. And if you go to the next slide, we started in April with, okay, great idea. What are all the things that we have to put together to make this happen? And there were a lot of them, and we were probably ambitious of when we thought we could get them done. And here they're all in gray. Green is going to be completed and blue is going to be in progress. So if you click to the next slide, we recently updated this timeline, and the take-home message here is not the specific details of each one, but there has been a green wave. So a lot of these things have been completed. A lot of them are also blue in progress, but they're nearly done, or I just didn't get the slide updated in time, and somebody will say in the comments, no way, that's already done. So really want to acknowledge the work within the entire team. A lot of different people worked together, aligned their work plans, and got a number of things done that's going to enable some of the really exciting interop that Brian is going to show in the next slide.
Great. Hey, Jack, can you hear me okay? Okay, awesome.
Yes, I can.
Right. Okay, thank you. So yeah, I mean, Jack showed, a really extensive list of all the technical tasks that this group was working on over the last, 7+ months. And, I just want to underscore, this was a huge team effort. It was a lot of effort and time and energy that went into aligning everyone's work plans and collectively pulling in the same direction. So I just want to, first of all, thank everyone for all their hard work in this working group. So there's clearly a lot of technical tasks that got done over the last, 7 months plus. But what did that actually, yield in terms of high-level functionality? And I'm really excited to see, what came out of this technical work. At the high level, we have examples of search result handoff working over PFB, right? So we have two portals. This is the NHGRI Anvil and NHLBI Biodata Catalyst portal. Sorry, there's a typo there. Those portals running on the Windmill portal software from Gen 3 are able to produce PFB and hand off search results to workspace environments, multiple workspace environments. And so this includes the Terra-based environments, as well as Gen 3 and 7 bridges. What does that mean for a researcher? Well, there are over 400,000 subjects that are accessible through these portals. So that's a huge collection of data there. In terms of data access, our work on DERS, and as Jack mentioned, we actually had a really great collaboration going with GA4GH, and were able to propose changes that were really helpful for us to the GA4GH API and get those adopted as an official DERS 1.1 version over the last year. And thanks to the work of University of Chicago and other folks here, we now have 4 DERS servers making data accessible on Anvil, Biodata Catalyst, CRDC, and Kids First. These are running on the Gen 3 stack. And that means that there's approximately, and Bob or others can correct me if I'm wrong, the number is probably bigger since the last time I counted things up, but it's about 6 petabytes of data are now accessible through this GA4GH API standard, which is amazing and exciting. And then as far as auth goes, I think we made more progress collectively than we thought we were going to in the first year of this project. We actually are seeing RAS being adopted for authentication, and that's being rolled out right now. So it's really exciting to see a GA4GH passport-based system being used across various stacks now. Now, if you go to the next slide, apologies ahead of time, whoever's driving the slides. I'll tell you when to start the video. We're gonna try a video and hopefully it'll work. But I want to take a step back. We oftentimes talk about these abstract technical tasks, or we talk about these high level bits of functionality, but what does it actually mean for a researcher? So let's walk through a typical researcher flow that is now enabled by these key pieces of interoperability tech and standards that we've been rolling out. So in this flow that I'm going to show you in the video here, as a researcher, I'm going to find data in a portal. I'm going to take that data search result to a computational workspace environment via that PFB handoff mechanism. I'm going to pull in an analytical workflow from DocStore via the GA4GH TERS API. I'm going to then run the analysis in the cloud using that workspace environment. And that analysis will actually in real time pull in the data that's needed over the GA4GH DERS API. So in this particular example that I'll ask to start in just a second, we use the Anvil portal, Windmill portal by Gen 3. We use DocStore for pulling in the workflow, and we use the Anvil workspace environment powered by Terra. Other combos are possible. I mean, that's the whole point of this interoperability. But I do want to show you this now. So let's go ahead and start this video, and hopefully I won't run too over on time today. Okay, great. It seems to be working. So in this case, as a researcher, I come into Anvil. I look at the data browsing portal. I'm going to log in. And for the purposes of this demo, I'm using my own personal account. It has no controlled data access. This is all public data that I'm going to look at. So now that I've logged in, I can go over to Explore. I'm just seeing the public data sets. I'm going to choose 1,000 genomes data and maybe narrow it down just a little bit more by looking at no data field for sex, for example. And that gives me about 700 subjects that I click the button to export all to Terra. A PFB is created in the background that represents my search results. I'm going to take it into a new workspace environment just for this particular analysis. So I'm going to fill out some info. I'm going to create that. And now Terra is pulling that PFB and creating a representation of the search results with pointers to data files in the background. And eventually that refreshes, and I'm able to go ahead and see the data model that comes over. It includes program, project, sample, sequence. In the sequence table, there's a bunch of data, including a VCF file that I'm interested in running an analysis on. I'm going to look over here, and you can see those DERS URIs for the GA4GH DERS are now interpreted correctly by Terra, and Terra is able to understand what a DERS URI is and how to access that data on my behalf. That's really an exciting development to see. Okay, so I've got my data model that's been exported. Now I'm going to bring in my analytical workflow. I know a workflow exists in DocStore that I'm interested in using. It's just a simple MD5 sum workflow, which is just kind of doing a trivial calculation on a file. So I'm going to click on that. I'm going to look for it. Here is my workflow that I created, and I'm going to send it back into my workspace environment. Again, this is happening over GA4GH TERS. So again, we have another API standard here for handing off workflows to a workspace environment. And I've just imported it into that workspace environment. Now what I'm going to do is I'm going to parameterize my workflow with the data that I selected. I'm going to look for the first VCF file that I was interested in. I'm going to wire up the input for this workflow is going to be that DERS pointer to data over DERS. and I'm going to hit OK and put that in the input field. And then I'm also going to specify an output field to collect my, in this case, just a checksum. And once I've wired the inputs and the outputs together, I can go ahead and save that and ultimately click the button that allows me to launch this. You know, I will say that I'm just doing one VCF file here, but in environments like Terra or Seven Bridges or other workspace environments, you can run on thousands of samples simultaneously. And so you can see here, it's been submitted to the cloud, it finished running. Now I'm gonna go back to my data model, and I'm gonna look in my original table, and there we go. The MD5 sum has been calculated and deposited back in this table, and I can get a little preview of that and see what the checksum is for that file. So that was kind of a whirlwind tour here of, next slide, please. A whirlwind, okay, great, yeah, we're in the right place. You know, it was really kind of a whirlwind tour of you know, what does this interoperability work look like for a researcher? And it looks pretty simple, right? It's a simple flow and it's easy to do, and it just works. But it actually is a lot of work went into that behind the scenes to make that simple. And so I think that everyone on this call should be, you know, proud of the work that we're all collectively doing to make that look and be simple for a researcher. The other thing I want to point out too is that our interoperability efforts are not in isolation. You know, through, for example, GA4GH, we're seeing others adopt the same set of APIs as we're adopting in NCPI. And for example, last month for the GA4GH plenary, we had an interoperability demo with multiple groups, including 2 of the stacks from NCPI. So Terra and Seven Bridges participated, along with a Canadian stack called DNA Stack, and the Elixir cloud environment over in Europe. So what we're trying to do here in this demo is take the same workflow, different flavors of the workflow, but the same intrinsic workflow, send it around to different compute workspace environments, run the workflow, pull in data over DIRS, and report back the result and see that we could run the same GWAS analysis across these very different environments, but using the same set of GA4GH APIs. And ultimately, that demonstration was very successful, and it's exciting to see NCPI you know, software stacks participating in these more sort of global interoperability efforts. And you know, it's because we really adopted several of the GA4GH APIs that really allowed us to participate in this. So next slide. You know, I would say this is really kind of the tip of the iceberg, and you know, we're looking forward to continuing to work, you know, on interop within NCPI, but also, you know, for me personally, I want to look towards the broader community as well. So, you know, in the long run, I think the broader sort of vision of interoperability in the future is, you know, not just really helping NIH systems through NCPI, but really opening ourselves up to global interoperability. Again, longer term vision here. But you know, the standards we're adopting now, I think, are setting the stage for us to have a researcher come into these workspace environments using their passport as their identity, to be able to search across many different environments, to be able to access data, you know, globally in different locations, pull in compute algorithms like workflows or notebooks, and then to actually trigger compute on the appropriate cloud in the appropriate place to minimize costs and ultimately collect results back that they can share, you know, with their collaborators and produce publications and share that with a wider community. So next slide, please. So that's, you know, it's great to see the progress we've made, you know, just in the last 7 months to a year, right? I think we've made some real tangible progress here. And it's great to see kind of us setting ourselves up well for future and more sort of global interoperability with more and more systems. But obviously the work isn't done. We have a very productive year, but there's many other sort of additional existing challenges and new challenges. Some of those are on the technical side, some of those are on the policy side, as Bob's talk presented earlier. On the technical side, I think, you know, one of our key questions is how do we get more systems to adopt these APIs so we can interoperate with more portals, for example. How do we leverage RAS not for just authentication, but also authorization. How do we find data? How do we do search and discovery? How do we find data across systems? Jack mentioned, the needs for common metadata models across systems. How do we accomplish, compute in the right place with the right workflow? We heard Adam and others talk about, the sort of challenges of having workflows in different flavors, the challenges of data locality and egress. And there are more, and we're gonna have a road mapping session a little bit later today that I know folks are already adding to the fun retro board. In addition to these technical challenges, there's of course policy, and I think Jack wants to jump back in here a little bit to talk a little bit about the policy challenges as well.
Yeah, so I think the table that Bob and Stan showed in their group really shows that how complex and evolving of a landscape it is. Even though it seems conceptually straightforward, I think the more that we drive forward here, the more that we're going to run into these different blockers and have to figure out these different ISAs or other trust relationships or things that make this possible. To the point of production, it's also adoption. So we were talking this week within the Childhood Cancer Data Initiative with researchers at St. Jude. So they hadn't been involved. Like how do we bring them into this group? Because some of the things, we're working on the same problems in this group that they're working on in a separate effort. And how do we bring those things together and get more people involved? Allison Heath brought up a really interesting conceptual, philosophical point about reproducibility in the knowledge life cycle, which I think we may talk about tomorrow or in this afternoon. But how do we deal with things that expire over time? And the discussions that were highlighted by, especially the CWL Whittle discussions this morning, these usually come up every week or two for me. I don't know what's in the air. I assume this meeting and ASHG, I've been in at least 3 deep discussions about this per day this week of, what are we going to do? How do we guide researchers to the right place? What about bringing things over? And I know Mike and I are going to lead a breakout on Monday about this. But again, the more that we're able to drive this forward, the more that people realize, oh, cool, we can do this now. What about that? And I think that's good. That's a nice outcome.
Right, we are a little bit running over time, so I'm going to stop Brian and Jack here, but I'm sure they'll take questions in the chat if you have any questions, and we'll have more time for discussion this afternoon.
Yeah, sounds good. Thanks.
Of course. So our next working group is the FHIR working group, and that is led by Allison and Robert. And so Allison has already taken the microphone from me. And so we'll pass it to her and let her show us what they've been working on.
Awesome. Thanks, David. Yeah, we're going to be doing a couple demos today. Hopefully live demos go well. So I'm going to talk a little bit about what we've been doing with the FHIR working group. I think my hat is a little bit about how I felt about clinical data back in the day, and also any discussion I ever have about metadata versus data and what does that mean. But I think slowly, I can flip it back because I've also had lunch now. To happy Mr. Meeseeks. We're going to talk a little bit about what we've been doing for the last 7 months, and it's been a really interesting experience, I think. So I was kind of thinking about energy barriers and how, sitting on a roller coaster and you're looking up and wondering, this is a really long climb and how is this going to work and what are we doing? I think this is really kind of feeling in March when kind of started and said, picking up steam in kind of the EHR space. What does that mean for research? And kind of prepping for this meeting, looked through some of the minutes, and I thought it was very interesting to see the very first 2 things, even though we hadn't really laid it out, is looking at leveraging FHIR to enable interoperability and how to break down silos between the different resources. And it was kind of a new idea of using FHIR for this type of work. So in April, it looked like this was actually really happening and that we should give it a name. And so we started off with this idea of something called Project Forge. And really this was to get everyone together, think about different ways that we could leverage FHIR, but also think of really collective learning experience of what is it? I think when you look at our original FHIR 101 guide that we made a while ago, You know, there's a lot of, I've heard about FIRE. Is it a database? Is it a data model? Is it rapid oxidation? And then every other pun that you could ever make about FIRE, I think, occurred. You know, you run through it, and I think if you're a developer, it looks like one thing. If you are a researcher, it looks like another thing. If you're a data modeler or, you know, someone, and I think there's a lot of aspects to it, and we'll talk a little bit more about that later. And yeah, I think the first May to July, looking through the minutes, it was actually a lot of what is FIRE, what's a resource, what's a structure definition, what's a profile, how do they relate to the different use cases that we're interested in, and you'll see, you know, we kind of went through this process with a lot of different people kind of coming in and out of the calls and started, you know, I remember every call starting out, you know, who's new, introduce yourself, what are you doing? And I think we heard this morning a lot of interesting things happening with the genomics, but still we need a need to really link it to the clinical data. And so we started deploying some of the servers. I think there's nothing better to actually try something than to try something. So we set up the different dev servers. We started getting people on board and started also testing this kind of hypothesis around FIRE of how standard is standard. Can I really develop an app and point it to any FIRE server and expect it to work? And I think the answers are some apps, yes, some apps, sort of, some apps, maybe not. But initially just browsing the data, creating a dashboard, and we'll show a little demo of this. It was surprisingly easy. We could point it to open access servers, we could put it to controlled access servers, and FIRE is self-describing enough that you can actually build applications over it to view the data. And so we went through this process where the existing things in FIRE that were in the research space had really put a lot of constraints, kind of taking what we've done, I think, traditionally of what's the data model? What are the common data elements? How do we put them in there? What do we name them? And we took a step back, I think, right around the August timeframe of, do we need to bring any of that? Can we just start with the base FIRE resources, what's already kind of been in different maturity levels, and actually load all the data we had in hand at that time, which is the PCGC data, which you heard a lot about this morning, as well as the Centers for Mendelian Genomics data. And I think the answer is yes. You can put all of this data in FIRE, find places for it to represent that are reasonable. The thing is it starts turning in a lot of things are what we call observations in FIRE. So you can make an observation about patient, observation of a specimen, observation of a file, and I'll show you kind of a little bit of what that looks like. And it becomes this question, is that useful? Sure, you can throw everything into observation, but part of what you need is pulling out things that are important to exchange and run our different tools and things like that. So after we went through that experiment and found out that indeed we could get a lot of this data, all of it at the time, really in there, we started looking at what FIRE calls profiling. So you take something like an observation and you add constraints on it. So it's still an observation, but you can say something extra about it. You can say this observation uses the human phenotype ontology. This observation is a family relationship. This observation is XYZ of whatever you think is important. And so, you know, it's kind of this concept of some observations are probably worth more notice, you know, based on the research objectives than others. And we started this process on GitHub where people could file issues, provide their use cases, and you can see these are some of the high ones that came up. I'll demo in a second about phenotype, disease, family relationships. And actually even how do we put in maybe DERS references into FIRE as well. And you can see there's actually quite a lot of conversations about some of these family relationship, a lot of comments and other things. And we encourage anyone to go in there, kind of label the use cases, understand what we wanted to do. All right, so that brought us to that point where we created this initial FIRE setup. We added some of the initial profiles, and then we start asking, what does that look like? So I'll start, hopefully this isn't too tiny. So this is the GitHub page, and this is kind of where we started of having FIRE 101, and again, kind of the different repos and stuff that I was just showing. You can see the issues there, all of that in there. And just this last week, we created an implementation guide of that. So you can see that this is publicly hosted as well. Not much description yet. I think we need to fill out different content and other things. But what I wanted to show here is the different table of contents that I was talking about. So you have things where you're already bringing in some of the fields that are defined by the US core, the definition that of the EHR, race, ethnicity. We can just pull those in and reuse them. Then we can add things like age and event, and then some of these structure definitions or more special observations that we really want to note in our systems have been vetted or validated somehow. So phenotype, family relationship, etc. And so you can drill down and look at that and actually look at different examples and other things. And these are kind of the human part of FHIR, of how do we document this? How do we understand what those different things look like? How do we provide examples? So if we go over to more of the computer part, if you go to these servers that we set up specifically for the working group, and these are actually accessible to anybody, you can go and, for example, our favorite word, metadata, and look at that. The FHIR servers are actually self-descriptive, so they can tell you about their capabilities. What does this FHIR server do? What does it serve out? And you can start understanding and acting on that accordingly. So that app that I was showing you the screenshot, here's the dashboard that's living on here. And now I'm moving over a little bit to a controlled access version that has the CMG and the PCGC data loaded in, and kind of linking towards how do we get developer access across this. But you can see in here, you have like NCPI phenotype, its base type is observation. We have DIRS documents. You can see there's 50,000 of these specimens, research subjects, patients, diseases, etc. And then you can kind of drill down and see the different information of, for example, the family relationship, or actually the phenotype. So you have the 50,000 here, and then you can see different high-level roll-ups of this. And if you go and you click in, you can actually drill down to the different roll-level ones, see what specific patient those are attached to, etc. There are 50,000 of them, so it's actually probably gonna take a little while for that to load. So I already have it preloaded a little bit. I was kind of hoping to show a phenotype, but maybe we'll just let that load. So you can see here as an example of what maybe DIRS might look like in there. And you can see it's just JSON, right? At the end of the day, these APIs are returning you different JSON information, and you can put as much or as little in here. One of the first iterations, we actually had all the MD5 sums, the file sizes, etc. But then the question is, do we really want that replicated? Maybe it'd be better just to have the DIRS URI here and maybe a little bit of information about, you know, maybe where that came from or who authored it. So you can see here, here's a DIRS URI pointing to an Anvil location for, I believe one of the CRAM files for this. And you can see within the same server or the same standard, we could have one of the Kids First CRAM files similarly there, and kind of writing off of the work that Brian and Jack were showing, have a DIRS endpoint here for the Kids First. So you don't have that, but it links back to the specific patient identifier, for example. And we can figure out kind of ways of bringing that data in. Last thing I wanted to note is, if you look at here, you can see all the different entities. So we have 50,000 phenotypes, we have the patients, but we actually have 2,000 some observations. So what other kinds of observations? And this is where, really you can put anything. So we were playing around with, can you put the sequencing experiment information in FHIR? So here is one, it has those document references referring to the different DIRS endpoints, but you could put in some of the metrics of the insert size, the depth, kind of other types of information that you might want to make indexable, searchable, that you could pull. And I was reflecting on this morning's conversation with BCFs and what are contained in there, what are things. So these are kind of areas I think we could explore where it's, FHIR makes it super easy to bring in a lot of the clinical information, but there's actually structures and stuff to bring in others. Okay, so that's great. This has really helped, I think, the developers to browse through data and for me to kind of show a little bit about there, but really, I think what gets exciting is once you have these APIs, you can start creating quick apps. So we actually built this in 48 hours in the last 2 days. So it is super prototypey. This is using Dash, which is kind of Python's answer to Shiny a little bit. And this is querying against the endpoints that you were just seeing. And it has kind of broken it down by PCGC and CMG. Let me make sure this is big enough that people can see it. And on this graph here, we just have the phenotype codes, and then each one of these bars here represents a patient. And the first thing you'll notice is that PCGC seems to be all the same. What's going on? And it's actually because PCGC does a full phenotypic profile, and we have a lot of negative observations. So we are including both negative and positive observations about the phenotype. If I remove the negative one, we can see this is probably more what folks are used to looking at. Each one of these is a subject with these phenotypes. And you can see across PCGC and CMG, there are many that have similar overlapping phenotypes. And this is part of why you want to bring these things together. And I can continue. We just did some kind of quick ones that everyone expects to be able to do, like gender, etc. As I hover over them, you can see, hopefully, if it's not too super tiny, that CMG, for example, is calling this phenotype abnormality of the face. Over in PCGC, dysmorphic faces, but they're mapped to the same phenotype. So you have this ability to bring in both the source text or what people had denoted, and then add a layer of different codes and standards over time to really be able to bring it in and be able to analyze. And being able to start looking at these in aggregate at the patient level, et cetera. So that was kind of the quick application demo view of looking at this higher, and then I'll turn it over to Robert to show a little bit about what you can maybe do on the individual patient level, and I'll wrap up our update as well.
Great, thanks, Allison. I'm going to collaborate your screen sharing here. Hi, everyone. So I'm really excited about showing this work off as well, although of course I can't see because the Zoom bar is in the way. Second. So I was just doing, you see the search page here, I was just scrambling at the last second to try and figure out some search parameters to use. But it's, once again, using RESTful APIs is amazing. So this is an example of Shiny app that I threw together a while ago when we first got the data there. And so what we're going to do here is we can actually go and take a particular research subject from the database that Allison was doing, and we can go get and run against the Monarch API, checking to say, hey, for this phenotypic profile, what are the top diseases that seem likely? And so it's really awesome, this integration then we can start doing with other tools. So I'm going to load in set this up quickly, just load in my cookie to access the data. And I have this one preloaded, it immediately runs. And so Allison was asking me, hey, can we slow it down a little bit? Because that's how fast it is from getting the authorization loaded to actually loading the information for this particular patient in the server and querying the Monarch API. It all loads almost instantly. And so you probably couldn't even tell the delay from populating this UI and having it already go to fetch results from Monarch API. So here we have this list of source, maybe I should make one bigger too. This list of source HBO terms here, right, is coming, these are NCPI phenotype resources from observation for that subject research study. And then list here, and you notice you might see on the other side, we've got some positives and negatives depending on the status they were assigned. So as Allison mentioned, the Kids First team does this really robust negation status for a lot of different phenotypes. It will also have listed from the Kids First source some conditions that were defined about diseases the patient did have. And so we can kind of compare that, what was listed in the original study, with what the Monarch API suggests about potential diseases. We have a sort of similarity score. We have number of phenotypes it matched. And so one could also then use this and use the Monarch APIs again and get target genes. And so now we could go very quickly from a phenotypic profile to some potential genes to test. And with the DERS alignment as well and all the workflow pieces, you could actually go very quickly from getting this clinical and phenotypic data into turning that around to a specific gene test targeted towards the top maybe 5 genes of interest or something along those lines. And so this, once again, was really simple, very easy to pull together, once again, using this quick, Shiny to do a quick demonstration. And you could even see too, if one was interested in saying, well, were there any other patients that actually have, any of these same diseases that were listed? And so we can actually very quickly, once again, run a query. This is just FHIR. This is using some of the reverse chaining they have, and say, hey, give me the list of all patients that have this particular NCIT term. And so that very quickly we got 41 participants back that have that term listed, that disease listed. And so now you can do that to expand your search to look for other similar patients or study participants, and it's all really powerful and runs very quickly. And I'm not an expert on writing against APIs and querying against APIs, but I was able to figure out in no time. Cool. So I think it's super thrilling to have this, and I might not play this. I can't find the button. We'll walk through the slides here. Cool. So we talked about, yeah, so what is FHIR? And one of the things, it's this framework for clinical data interoperability. And I think one of the areas that we've really come to is, while there is a lot of work that we described that we've been doing, like frankly, it's just like any of these other frameworks that we use all the time to build platforms, right? Because it provides these opinions where it's really important, so we don't have to keep solving the same problems over and over again, but also provides that flexibility so that we can customize it to the work that we're trying to do. And some examples here with Gatsby, Django, and Kubernetes, right? These very commonly used frameworks that really empower the work that people are doing without really constraining it. And so this idea, right, a framework within a framework. So part of the challenge that we ran into that Allison talked about before, one of those challenges is FHIR is actually sort of three things, right? It's a data modeling language, it's a data model itself, and it's an API. And so you hit sometimes you can hit that documentation and it's just overwhelming. But really we can focus on this flexibility piece and break that down even further over FHIR to say, well, really, we get these schemas, these structure definitions, we get sort of the community buy-in about what's important, how should we model things. And the HL7 community is really large. And then also we can do some of these profiling and constraining the resources down to match the type of work we're trying to do. And so we can make statements like, hey, if you want to be able to use HBO terms against the Monarch API, you need to provide a resource that looks like this, that actually has these HBO terms in it. And that's great, and that's really helpful for everyone. And so I think it's super exciting to kind of reach this point where we're like, oh, cool, we kind of get it now. And we're actually able to really quickly build these apps and use other APIs and connect across the way. So yeah, once again, the retrospective here, right, this climb, all the work we've been doing, really over the last 6 months and some change, right, is trying to understand the framework, why it has some of the opinions, what are the advantages and disadvantages that we might encounter using it, so that we can really leverage that framework to do exciting work. And so once again, these tools that we're showing, in 48 hours, Allison and I think Mean put together that dashboard, or the fun little dashboard, right, with the distributions. Like, that's amazing. Like, and that's the kind of thing that you can do when you have an API. And if we want to pull a new data set in, it's not a big deal, right? It's the same flow of data. You could pull EHR data in and probably very quickly do the same type of thing with billing, ICD billing codes instead. And that's just really powerful. And so once again, right, there's going to be challenges ahead, right? Things that throw us for the loop here. But at the same time, right, having these tools and libraries and apps, the more we build out, the more synergy there'll be, and sort of the more we get by continuing to follow through with this. So with the roadmap moving forward, right, we're going to continue to document those current best practices. Allison showed the IG that we have together, and we want to move from that Project 4 model to really what we think will be the core NCPI FHIR model. We want to figure out some new cases, right? Cancer research is an important one. The clinical genomics aspect is really important to some of the AMBL work too. And really, whatever use cases there are from the platforms, it's really important that we get these voices heard and we're able to build towards what everyone needs. And once again, right, trying to identify those continued unmet needs so we can build the tools and to really build the synergy more and more, right? Especially intake management, availability, interoperability. Those are all really critical steps here. I think that we can improve a lot from the world of, well, I've got this CSV with a PDF data dictionary that I'm going to drop in a bucket, right? I think there's a real opportunity to transform that process into something much more modern and exciting. Yeah, and so we really want to make them available. And once we get some of this loaded, we can make these tools available. People can go develop their own apps in these same environments that already exist to doing the workspaces. And so I think that's really exciting too. And then of course the community engagement, because you can build, anybody can build and apply apps against a standard API, right? And that's super exciting. So we've got a bunch of people showing up. This is sort of contributions. over the NCPI fire model here. We've got some big peaks in there. We got a lot of good work done. And we've got some real special thanks for demos today. Go out to some of our teams, the Ample team here with Brian and Chris and Eric and Katie on our side, and then the Kids First team with me, Nick, Shane, Natasha, Avi, and Alex on Allison's side. And so I'm really excited. I hope this has shown you guys something to be excited about. And I think I'm really looking forward to tomorrow getting a little bit more in depth and hopefully about the use cases and trying to sort some next steps out with everyone. Because I think we're really close to something. We have done something really cool, and I think we're really close to making it available more broadly.
Great, thanks so much, Robert and Allison. So we're running a little late. So again, if there are questions, then you can feel free to put them in the chat. But otherwise, we're going to move forward with our last working group, which is the working group that I'm a part of, the Outreach and Training Working Group. And we are chaired by Anton Nekritenko and Ashok Krishnamurthy. And so I will get the slides set up here and hand it off to them.
Thank you, David. According to schedule, I should be already done. So I'm quick. So I think, well, you are a part of this working group. And so I think this workshop is a testament to your efforts.
Oh, well, thank you.
So goals of this working group is enable cross-pollination between NCPI, between projects participating in NCPI. Also, we are going to take more active role in development and maintenance of NCPI portal. Right now, it's essentially part of the Anvil portal. And so we're gonna focus more on that. And our two immediate goals is providing a catalog of data sets available from all these resources. So you can view this as a kind of a library catalog. So you can have bird's eye view of what's available, and also trying to provide centralized place from which tutorials for all 4 platforms can be accessible. Next slide, please. So you just heard a lot about FHIR, and FHIR is one of those things is that nobody knows what it is until you've tried. And the Kids First group, and in particular Allison, they have developed a wonderful tutorial, which you can see at NCPI GitHub page. It's called FHIR 101. So what we wanted to do is make it even more sort of for common people, more accessible. And we created a more diluted, if you say, version of that. You can try this at this URL. It's using Google Collaboratory. And in this particular case, what it does, it's accessing FHIR server, which dbgap runs for some of the example queries that you can run through this. And so this is going to be also listed at the NCPI portal. Next slide, please. This is a prototype of the global data dashboard. If you look at Anvil portal, you will see the Anvil data dashboard. So essentially, we're going to use the same framework to try to list data from all 4 platforms. So at this point, this is prototype. It's not yet on NCPI portal. It's in one of the branches on GitHub, but it will be there. And so it tries to list data sets from all 4 portals. The way it really works right now, it works out of a static Excel spreadsheet, which was given to us specifically by Garrett. And it's using dbgap FHIR server to pool the relevant information. Next slide, please. So this is sort of a magic of this dashboard behind the scenes. One problem with well, it's not a problem, it's a caveat of the current version of that is that it only lists platform-specific data sets which are in dbgap, but not all data sets provided, contained within platforms are in dbgap. As I mentioned, it's really a static spreadsheet at this point, and it uses some of the dbgap interfaces, including the FTP XML and the FHIR API. And it was actually, this was one of the great things about this was establishing the relationship with the dbgap FHIR group. And this was so this contacts was established with help of Valentina, and they're really a pleasure to work with. It's they want to change the API per our requests, and in general, they're very helpful. And the plan is to use the GA4GH discovery API in the future, because obviously operating from static spreadsheet is not a good plan. Also, what we want to do is we want to list all tutorial materials for 4 portals. This is an example of how we do this currently in Galaxy framework. We have a Galaxy tutorial training framework. And so if you show the next slide, please. What we ultimately want to do is to start with a simple grid where for certain types of tutorials, it will list which portals have these tutorials. So various ways in which users, especially naive users, can get idea how to start using these tutorials. So again, to wrap this up, we have 2 immediate goals: the global data dashboard and making all tutorials accessible. Thanks.
And you've been there. That's great. Thank you so much to all of our speakers. And so we are only a little bit over time, so that was great. Good effort on everybody's behalf. And so we've got on our schedule now for a quick break, and so we'll take a 13-minute break, and we'll be back here at 10 after the hour. And then we'll be looking at the Fun Retro board and starting some discussion. All right, so I have 3:10. So let's go ahead and move into our last session of the day here. So I'm going to pass the baton and the microphone over to Allison, who's going to lead our discussion today.
Actually, myself and Brian. So.
Hey, Allison, I'm back.
Hey, Brian. Yeah, just want to make sure you were there. So actually, I had. Okay. I am sharing the Fun Retro board, and David, if you could link that in the chat as well so folks have it readily available. So yeah, thank you, all the different working group updates. I know across the board people have been working really hard, and part of the goal of this workshop today was to kind of talk a little bit about where we've been and start hinting at where we're going. And so we thought we would do this exercise of trying to bubble up different things towards interoperability and towards what we heard today that have come up. So hopefully everyone's on there. And you'll see, if you've never used Fun Retro, it should be pretty straightforward. If you want to add something into a column, you can just click the plus and type whatever fun stuff you want to type in there. Or if you don't want to, you can delete it. And you can switch layouts, all that kind of stuff. And so I thought we would start. I'll do kind of the first part, and then Brian will help. So the second part of, maybe the first part, really thinking about some of the different data sets or initiatives that we feel are driving the interoperability. So we've kind of put some ideas here. But if folks want to take a few minutes and kind of think about other projects, other data sets, other drivers that are kind of coming up over the next, I'd say 6 months, because, I think there's always a delay between when data kind of arrives and when we can add features to it. And then the other thing I'll note, part of this is you do have votes. So you get 6 votes. And it's across all of these topics. So I thought we'd take a few minutes and people can kind of brainstorm and think about that first column there. You can vote now, or we're also going to vote later as well. So
and Allison, so just to be clear, you want us to focus in on, and I don't know how many minutes we should do this for, but focus in on the data sets and initiatives driving interoperability kind of now. If folks have other ideas for, the technical tasks that you have in 6 months and 12, that's fine. But we want folks to focus in on the data sets, spend a few minutes, adding additional ones, voting for data sets, that sort of thing. And then we'll discuss and then repeat the process for the sort of blue column and purple column, or well, pinkish column and purple column. Is that right?
Yep, perfect. And you left a little open-ended. You know, a lot of the ones here are data sets, but you know, initiatives. I see CFDEs showed up there. The Childhood Cancer Data Initiative. Some of those other ones, and we can kind of discuss as well.
Great. Okay.
Jack, you're asking data sets split between ICs or people want to evaluate other workspace? I think both. And if people want to put some notes about, you know, why they're drivers of interoperability, I think the other thing you can do is you can comment on it. So if you wanted to add something about PCGC, of course, now my screen is frozen up. Why is that?
Oh, yeah, I see the little dialogue cloud. If you click on that, you can add your comment.
So if you want to
that's great. And the other thing to note too is you have 6 votes. If you kind of change your mind, you can untick the vote for something and put it on something else as well.
Yeah, and I see Allison Leaf was asking about what are we voting for, which is always a good question to ask. You know, I think on the roadmap part, you know, maybe we are voting for the priorities. I think for the data set part, it's more, you know, have you heard interest for this? Is this kind of a priority to support it for your, you know, researchers or projects that you've heard kind of coming through? And I think that's why with the data sets initiatives, we were actually, you know, maybe not voting so much as trying to bubble up what folks are thinking across the board here. I agree with the roadmap votes, and maybe we should have made 2 boards, but you know, such as organizing these things. All right, so
I have to say, Allison, this is working really well. There's a lot of activity. There's a lot of you know, data sets and projects that have been mentioned, a lot of votes. So I'm definitely liking the fun retro.
I think I should be able to sort it. All right, so give people one last minute to vote for data sets, and I guess we will allow people to unvote when we go to the roadmap section. You can, but kind of thinking about which ones are ordered by vote. All right. So interestingly enough, right now, kind of at the top, we have MISC-C, the Cancer Data Aggregator. Oh, Cancer Data Aggregator just outvoted MISC-C. You know, CFDE, CCDI. So normally how we do this is often if someone wants to speak up about kind of the Cancer Data Aggregator and CCDH, either who wrote it or someone who voted for it, about why you feel this is an important need in the interoperability space.
Yeah, I can just mention I ended up creating this one, and you know, again, I see a mixture here of data sets, and I see a mixture of kind of platforms and sort of projects that cut across data sets, which is great. But I put CDA in here, and hopefully Melissa Handel's still on the call. Maybe she can chime in here too. The perspective, I'm interested in how do we do discovery and queries across. This is something that, you know, within system interop, we didn't really address the issue of like, how would you do a distributed or federated query across these systems? And so CDA is looking towards building that infrastructure for the NCI. And the other question that's been brought up multiple times is kind of how do you move closer towards a harmonized, you know, data model, harmonized metadata model. And Melissa, do you want to mention a little bit about CCDH if hopefully you're still on the line.
Melissa's not here. I can say a few words.
Oh, thanks.
Hey, yeah, this is Sam Bolchenbaum. Yeah, so CCDH is trying to provide guidance over all the nodes and data resources as to how we can develop, you know, a harmonized common data model, but more importantly, how we can develop best practices across the nodes for everything from data ingestion to quality control to harmonization to aggregation and sharing. And obviously, between the nodes, which have all grown up at different times, there have been different ways to do these things and different ways have developed. So we're working closely with the nodes and obviously with the CDA group who's building a lot of the tools to come up with best practices. And we'll be doing this over the next year and a half now, trying to provide better ways for building interoperability and data sharing. Obviously, not every group is going to accept a model, and there's going to have to be a lot of give and take. And so we're looking for partnerships and for a lot of interest in working together and perhaps compromise where needed.
Awesome, thanks, Sam.
Maybe Brian and Allison, it's also worth pointing out that that some of these initiatives or data sets are sort of just now starting out, while others are more established, and likely that those two scenarios present different opportunities to build in either interoperability sort of ab initio versus working towards interoperability for around high value data sets and initiatives.
Yeah, I think that's a really important point. I noticed TCGA showed up on this list, and TCGA is, I think, a good 15 years old now.
It's an oldie but a goodie.
Yeah.
Do we want to talk a little bit about Miss C?
Yeah, so I think 10 people or so voted for Miss C. So does, I think I put it on the list initially, but I would love to hear from someone who upvoted it.
It would be good to hear about it for that, but for any of these, it would be good to see highlighted, like which platforms would it would need to be interoperable for that particular initiative, particular driving need.
So it's not clear to me yet. This is Stan, I upvoted it. It's not clear to me yet which platforms are going to get the MIS-C data, but we believe that some of the data will come to BD Catalyst, and I assume it'll also come to Kids First, given the nature of the data, which is data about kids that have, in a very non-medical way, kids that have an extreme reaction to the COVID. And it seems to me that it's going to be an important set of data, A, because it'll be very time sensitive. B, it's not going to be very huge, but it's going to be incredibly important because these are, kids manifesting kind of the worst the virus can manifest, which probably means something about a genetic link. At least that's from my crude assessment, not knowing anything. And three, this general inflammation process seems to be at the core of a lot of what COVID-19 seems to serve up to people of all ages. So to me, figuring out how we can keep this data and protect the data and yet use it across different, let's call them apps or tools on different platforms is going to be critically important, because understanding the phenomenology of how this inflammation manifests and through what pathways, from my completely uninformed non-biological perspective, seems to be very important.
Yeah,
So,
that's right, Stan. This is Valerie. I actually didn't put it down. I didn't vote for it, but I am involved in coordinating an interoperable approach to sharing the MIS-C data, which will come in through multiple protocols and other programs across the NIH and import, as well as Biodata Catalyst and the Kids First Data Resource Center will be involved. So leveraging the expertise of all your platforms and exactly what you just described, Stan, making it so that we know that this condition impacts the heart, obviously, and likely the neurological systems as well. So especially with the birth effects and cancer data sets that we have that are already impacting those systems. And then, of course, the associated data, immune profile data, and, heart data and other COVID metrics that will be going into Biodata Catalyst.
Yeah, and one other thing it'll drive us to make sure that we start to think about data outside of the data that we're mostly focused on, genomic and clinical data. But this will be imagery and other types of, assays, et cetera, is my guess.
and including, hi, Paul Aviak speaking from Biodata Catalyst, there will be in one of the 3 cohort that Valerie mentioned, there will be for the music study, EHR data coming from 29 different sites that will be linked to the registry data that is prospectively collected, linked to whole genome sequencing data. So it's a unique opportunity to work with all those different data sets in a prospective way and not just prospective after the fact.
There is also a driver of this that's one that it's there's a higher prevalence in certain ethnic groups, which suggests a genetic background basis, and it's a post infection sort of phenomena that may be mimicked, if we're not very lucky, by immunization. And so there's a real concern that if we're going to start immunizing millions of kids, even though this is a low frequency occurrence, it may be really, a small frequency of 1 million is still a substantial number of kids, and it's important to get this moving as fast as possible.
Maybe just like Ms. C, I think CCDI is also, a fairly new and emerging context. I don't know if somebody from the NCI would like to provide a brief overview for that, or if everybody's fully aware of it. I think many of us are upvoting it.
Is Jamie still on? She was on earlier.
Don't see her.
No, Jamie had to get off, unfortunately.
Okay.
So I'll chime in a bit on CCDI. I mean, this, and it's a little weird because the comments aren't showing here, but some people typed it right in the title. But this is an opportunity to really demonstrate interoperability across a number of different data repositories that today are not really connected. The Kids First data resource obviously is part of this whole interoperability activity, Target and GDC also, but bringing in things like St. Jude Cloud and bringing in, Sam Wolchenbaum's data resources at Chicago, Treehouse at UC Santa Cruz, for example. These are existing, some well-established data resources. We're not going to consolidate. We're not going to aggregate these data. We need to provide a way for researchers to find these data no matter which resource they're actually sitting in. So I think a discovery and then also remote analytics is going to be important across these data sets.
Excellent. And then I think the fourth one, and then we'll probably move on to the roadmap, but I see a CFDE is fourth behind it. And I think we have had some of the CFDE folks start joining some of the working groups, but if someone on the line who wants to just give a little brief along those lines as well, that would be great, although the description here is nice and descriptive as well.
Thanks. So this is Chris Kinsinger out of the Common Fund. And yeah, CFDE, we made some awards in September. Basically, there are some 12 to 13 Common Fund data coordinating centers out there that are dedicated to their specific Common Fund programs, Kids First being one of them. And so within the Common Fund, we're trying to connect the data between those data coordinating centers. And one thing we're doing is building a portal to search data across those different data sets from those programs. And then we're also looking at how do we make sure that these data remain available and sustainable even after the 10-year or whatever program ends. So we're working on that. And within CFDE, we have Kids First and also the GTEx data, which will be in Anvil. So we're kind of involved in NCPI, but also trying to figure out other ways to engage.
Awesome, great. So yeah, and I think looking at the kind of the list of things that came up here, I think there's, to Adam's point, some of the, high value data sets that have been existing a while, like GTEx and TCGA, I think model organisms are on the list there as well. Sounds like there's a lot of opportunities for, I think, further discussion. But since we have limited time today, I think we are going to try to use the last 30 minutes that we have together to really start digging into some of the roadmap issues. So I think similarly, the idea is now that we've kind of gotten a survey where we've been, so maybe something that's coming up, really thinking about on the NCPI front, either as part of a current working group, or if there's something going on out of band that is interesting, to start brainstorming, put them in the 6-month bin, which in my head is almost like these are things for sure we're doing, or the 12-month bin, which is a little bit more like, okay, these are things that we can do. It's not quite sure where they're going to be slotted, and hopefully, as we said, there'll probably be another workshop in 6 months where we can refine, but starting getting some of those things a little further down the road. And then the third column there, really new things, or things that we don't feel like we talked about today that are of a priority. And just going through the same exercise. Brian, do you have anything to add to that?
No, I think I tried to add some in where I tagged the, the working group that might take on it, or multiple working groups that might take it on. So if you're adding to it, you might think about doing that, or say, working group to be determined if it's a new area that's not covered by our existing working groups. But Allison, do you want to take, how long do you want us to take to brainstorm here before we come back and kind of do the same process of looking at the top votes?
I think we'll give people about 5 minutes or so to come up with things, and then a minute or two to vote.
Okay, great. So we'll go ahead and take that, go ahead and enter in what's missing, and go ahead and vote. And in 5-6 minutes, we'll start looking at the top ones then.
And yes, you can actually move these around. You can move them back and forth between the columns, if we decide that we want to do that. And you can actually merge them also if we think things are the same.
How do we do a merge, Alison? That I can see like 3 which look like a duplicate.
Anything in the 6-month roadmap, that's when we'll start, that I think is the same.
Yeah, we can maybe start with, are there duplicates, and then kind of merge them.
I find it easier to look at it this way.
Yeah. Ian, do you see any duplicates in the 6-month roadmap?
I see one that shows up in 6 and 12 and emerging.
Okay, across the board.
But all to do with search.
Yeah, okay.
I think the same is true for the accurate workflow cost projections.
Yeah.
Okay.
So Allison, what do you think? We've got 20 minutes left or so. Well, 22 minutes. Do you want to start with like the emerging concepts, or do you want to start with kind of 6-month, 12-month, and then emerging concepts? It looks kind of like the emerging concepts are things that are, most of these have things in the roadmaps, that are related to the emerging concepts. So what is your preference?
Start with the 6-month, Brian, do you want to do the top 2 or 3?
Yeah, that sounds good. Okay, so on the 6-month roadmap side, if you look at the top 3, let's say, outreach and training, code examples, how to do interop, that comes to the top. We've got this system interop and fire group, like how do we work together? Is it PFB or bulk fire? Is it PFB and bulk fire? What's the path forward there for sort of, sharing of, a bulk collection of search results or whatnot. And then the third item is, well, I'm glad to see this one, a revision of DERS. One thing that we ran into, in this project and other efforts is, how do you tell a person that's trying to access a DERS resource, this is how you get authorized to access this particular data object. So that's really what that one was about. I'm going to do a bonus here and say that the fourth one here was about RAS, which I think is, top of mind for many people on NCPI. So those are the top 4 for the 6th month. Does anyone want to talk a little bit about the outreach and training, the top result here?
I was going to comment, Brian, that the one you just mentioned, the way you described it almost sounds like an outreach and training exercise, which the question of how to get auth, right?
Mm-hmm. Yeah, there's 2 aspects. There's like how the human gets off, and then how the computer gets off, right?
From your comments, I was assuming we knew how to do the first via passport, though that subterranean, we know that that's not the case.
Yeah, and maybe this particular one is a little bit in the weeds because this is kind of talking specifically about how DERS would tell you which passport broker to go to. So that might be kind of not as broad of a particular conversation with everyone on this call, but it is one of those technical things that needs to be checked off in order to improve interop.
Yeah, but just to highlight, without going into the weeds, there are weeds to do with egress charges in here as well. So as a heads up.
But to your point, Brian, I think around the outreach and training component, my sense is that this is a thread across each of the platforms, including for even CFDE as well. And there might be some rationale for cross-coordination and or harmonization across our outreach activities, training activities, given sort of the scope of platforms and use cases that are being presented across the different groups. So that might be a really unique opportunity in a way to cross-leverage different training materials. I think some of this is starting to take shape, but maybe you could be a little bit more strategic about it.
Yeah, I totally agree with you on that. And Ian, I don't want to put you on the spot, but the work that you did, with your kind of, you did sort of vignettes for GA4GH in terms of how you would use multiple systems in a series of Python scripts that I thought was really remarkable and kind of points, towards this sort of code examples of working across systems. And so, that's speaking really very much to what Adam is talking about here, which is it's not just outreach on how to use this system for this data set, but I think many researchers are wanting to understand how they can span these now increasingly interoperable systems and see examples of that.
Yeah, I mean, I won't add anything to what you said, Brian, but the connection with what Adam mentioned, yeah, it's definitely there.
Mm-hmm. What about the next one? So this is 12 votes, System Interop and FHIR working group working together to kind of explore when are you doing sort of portal handoff, a search result handoff versus what can be encapsulated in bulk FHIR. This is something that, for me personally, I want to dive into this. This is my sipping eggnog by the fire, Christmas time, holiday time coding project, I think, when all the emails calm down. I'd love to dive into this, but I'm sure others on the call would love to dive into this as well and really try and understand how the two relate to each other, and are they the same? Are they different? They have the same use cases or different use cases. Any thoughts on that?
Yeah, so this is Jack. I totally aligned, Brian. Totally agree. This is probably one of the most important things that System Interoperation Working Group or NCPI in general can look at. And it really, they both seem really promising. And I think we all want to avoid setting up another situation where you have competing standards or competing workflow languages that can do the same thing. And, finding the path forward and getting everybody to agree on it, I think is really important.
I mean, I think we might want to line it up. So you've got, there's pairings here, right? You've got FHIR and PFB paired here, and you've got GA4GH search and FHIR paired up under the 12-month column. One might bring those three together, focus around what's the use case you're trying to do.
Yeah,
I think that's a great point.
And there's a big spectrum there across that idea of just looking for data, just the metadata pieces, being able to retrieve the actual full data, do you want to be able to do queries on someone else's platform to get pieces back? And I think that's one of those, the nuances there of what the different services provide, that I think it is really important to make that context, like you were suggesting, in as broad as possible.
But there's also, you have to bring within scope the the beacon use case, which doesn't give you much insight. In those sort of walled garden situations, all they're going to give you is, we're going to do the query for you, and we'll tell you a yes/no answer, because you can't look at inside those. I mean, that's a scarier use case and a more difficult use case, but probably will rear its head somewhere.
I want to just go ahead and move us on, though, because I am conscious of time here, and I know we want to get to 12-month and emerging concepts. So does anyone want to comment on RAS? I think maybe that's a non-controversial one, in the sense that, we all recognize RAS as being the auth N, auth Z, source of truth here, and extremely valuable service. But continuing to support it is, a very, very immediate term goal. Any comments on that?
I just want to say, yeah, I think it's been great to see RAS emerge. And to your point, I think we have a session on Monday to really dive into some of the details of that. So it's great to see this upvoted.
Great. So Allison, do you want to move on to the 12-month then?
Let's do it.
Okay, awesome. So the top one here, we've got a tie, and the top one listed here is high-level cloud scheduling, how to balance egress, compute cost, algorithm availability across clouds or stacks. I went ahead and I wrote that one. Sorry, it's too wordy, but I think this is a common theme that we're hearing across the researchers in the morning, in terms of, one thing being scary is kind of understanding what you're getting into as you do your compute across these environments, in terms of things like cost. And also there's a time element too. If you have to shift a bunch of data from one cloud to the other, versus the cloud that you're working on has the data resident and it's ready to go. So does anyone else want to comment on this one? I think this is, a very common concern, and chipping away at this is a high long-term priority for this group. Any other comments?
So Brian, I do have a comment. It struck me listening this morning that this is what you've written here is a very good encapsulation of what we have to deal with in the long term, of making the multiple clouds, each of which have their own, and I'll use my little air quotes again, their own operating systems, if you will, because we rarely go down to bare metal on anything, because that means we have to rebuild everything that Google's building and Amazon's building, et cetera. So we have been here before. This is the same problem we encountered when we had to deal with, figure out how to do Beowulf and cluster computing, and how to manage all of these HPC issues, like loading up the queues and making sure that you're using all of the resources economically, if you will. So in some sense, what we're trying to do is to rebuild some kind of general computing system that is across multiple clouds. But there's a confounder here, and that is all of the issues that we have around security and permissions and policy, et cetera. So if we're going to go down this route, which I think we have no choice but to do, I doubt that CWL and WDL are going to be totally reconcilable and sufficient for us to be able to operate and do the types of computing we ultimately want to be able to do. So that's the reason in the 6-month roadmap, I happened to put down a really ridiculous idea, but it struck me as not a bad idea, which is, why don't we get rid of all the security and policy issues and see if we can do some real interesting science on completely open data, meaning model organisms or something like that. And that way we can do real experiments, a lot of them, maybe even micro experiments, to really get a good handle on how do we start building the operating system for multiple clouds, if you will.
I would agree with Stan, and I just want to say, I think this is a whole separate workshop in and of itself. Truly. And it's a bigger problem, I think, than any of the projects represented in this group can take on their own.
Agreed.
Yeah, the tricky part is, as Stan, you were saying, like, how do we choose data sets that we could experiment with that would allow us to test certain aspects of this, rather than getting caught up on other aspects of this. So it's the isolation of particular issues and kind of chipping away at each of those.
Exactly.
It's
so we can
it's
make
a tough
faster
question.
progress. Yeah, but can
Yeah,
we make
yeah,
faster
exactly.
progress?
Yeah. Okay, any other thoughts on this one? I do want to spend a little time on we've got about 10 minutes left, so I want to spend a little time on the next one too. So this is actually, the next one is GA4GH Discovery Search and FHIR for queries. So this is coming back to Ian's earlier, question about, discovery, FHIR, PFB, like how do these all play together? And these are all kind of, sides of the same coin, or, well, not really a coin, but you know what I mean? Different facets of the same sort of question. Any additional comments on discovery search and FHIR?
Hey, this is Brian Walsh from OHSU. Apologize if somebody has brought this up already, but on this particular one, when I was listening to some of the presentations this morning, the use case, we all know there's a use case here. I'd like, in my ideal world, have a research project or two that is looking at discovery as part of their workflow, and try to have that alpha user or beta user for that, in simple terms.
Yeah, Brian, that's why I put that end user engagement under 6 months. Engage end users for search use cases. That's what I'm trying to hit on.
Like I'm going back a couple of years, the beacon idea is great, but I was hoping that we would be able to do metadata search across hundreds of projects on different platforms, and deliver a bundle, to use the DERS term, that could kick off a realistic workflow, etc. But again, not having to manufacture use cases, but one or two beta users that have a legitimate driving need for this would be an important part of it, if such a project existed.
Yeah, I think for me, you underscore something that I think is really important, and something that, in systems interop, we did early on, but it was extremely helpful to give us a guiding light, which was, making sure that we understand this sort of researcher use case and linking that to the work that we're doing. So, there's a ton of work that's in here. Like there's multiple years worth of work laid out for us here. But I think it's incredibly important to not necessarily just do sort of made up examples, but also to really root what we're doing in, even if it's open access data, but to actually have real scientific use cases. And that's something that's not necessarily reflected in the board right now, but I would say 2021, as we start up the next year of NCPI, making sure that we have, a shared backlog of these, some of them active, maybe some of them more speculative and in the future, but core use cases that can help speak to, we're developing this because we see researchers following this pattern. And I think that's incredibly powerful.
Hey, Brian, this is Dave Rogers. I just want to totally agree with you on that and say that advertised back in the 6 month one, there was a request to kind of take the use cases we have, the driver project we have, and refine them a bit so they can be understood by maybe a wider audience, and used as a pattern that you can do work like this or similar, just to really communicate what that is. And then once it's refined, put it on the NCPI site. Yeah.
Yeah, and in 6 months or a year time to look back and have that PI present what they discovered or how that fed into their overall project, etc. That would be you imagine the future in 6 months or a year, somebody's presenting.
Yeah, thank you.
Sorry about that, I'm breaking up here.
Oh, no worries. All right, thanks, Brian. Thanks, Dave. Okay, so it's interesting as we continue to go through these, the voting continues, so things move up and down. I just want to shout out though, there are a few picture items. So again, this is kind of coming back to that whole, conversation about, there's FHIR, there's discovery search, there's picture. I don't know if someone on the picture side, Paul or another team member wants to describe a little bit here. And then I think we want to cap it off with something that moved up on the list is tools and workflows. So let's talk briefly about the picture one, and then we'll move to the tools and workflows, since I think that's important too.
Yes, thanks, Brian. Paul Aviak speaking. Where within the Biodata Catalyst, where we have the Picture API deployed, which is an open source API that anyone can under an Apache 2 license, and definitely enabling access not just to the files, but to the variables with the Picture API. So anyone can search the distinct variable without the need to download the full data set. The same for the clinical data, but also for the annotated variants. And that's where we have the TOTMED today Freeze 5B available, so that it enables someone from another platform to access the annotated variant and the clinical variables, but only for the ones that are needed for the analysis, to avoid downloading the full file. So this joins also the issue of the reducing the cost and also the time to move all this information, because you're only focusing on the clinical and the genomic data of interest for an analysis.
Mm-hmm.
That's
great.
On the first topic. And on the second topic, related to enabling interoperability, and that's something that we started to discuss with Alison in the context of also with Kids First, of having interoperability between the BurgFire export and the Picture API, to enable interoperability between those two API standards.
That's great. All right, thanks, Paul, so much for that context. I
Hey,
do want
Brian.
to touch on one. Oh, yeah, go ahead.
Go ahead. Sorry. Well, sorry. I'm just wondering, we have a inherently kind of small group in this conversation, which is great. And it's been really useful in driving this forward. But I wonder, as we consider 12 months, like we have a very genomics focus here in the group that's attending. Is it worth considering how we reach out to people with other issues, trying to do sort of data integration across domains that may not be like inherently part of this small group?
Yeah, I see two farther down on the 12-month, the imaging data, and I see health data. And so I think there is an appetite and an interest to go beyond just here's your cram file, right? So I think that's a great point, Becky, for sure.
It comes through CRDC, but and I, the question I had in my mind was exactly that question. Is, I mean, CRDC has got proteomics imaging, et cetera, whether the other platforms are any have that diversity yet or not, because the same thing was striking me, not something we can necessarily answer now, but I think it's important.
Well, Ian, to your point, I mean, I don't think we do have that yet within Biodata Catalyst, but we will soon, and there's definitely an appetite for it. So given that GA4GH is having so many of these conversations around genomics, I'm wondering, where do the other conversations coalesce? And I'm wondering if maybe that isn't an important role for this NCPI group.
Yeah, I mean, with regard to GA4GH, I'm trying to work out the imaging example in DERS, because we can do that now. There are DERS objects that come back, which are images out of CRDC. And there's a whole bunch of questions related to that. So it's definitely beginning to be explored.
So I will just say, as a card-carrying toxicologist, if we could get to some kind of biomarker environmental data, or even find a place to have that conversation, I think it would really, I mean, it would be a hard conversation, but it would really serve these domains well.
Yeah, thank you, Becky, for that, broadening our context in terms of data types. I think that's really important to keep in mind. We have 1 minute left. So Alison, I kind of want to hand it back to you, because I see the 4 on the emerging concepts. I really take away the top 4 here, which by far have the most votes, as kind of like a really good sort of summarization of like the core concepts of much of what we talked about in the 6-month and 12-month. What are your thoughts on that?
Yeah, I think it's really interesting to go through this 6-month, 12-month, and then to see the emerging concepts. and you can kind of see data search across systems. As we talked about, I think there's multiple different layers. I was trying to put this fun trick or treat, trick and treat, trick X or treat thing in the Zoom chat. It didn't quite come across, but I think there's just many different layers to think about as people are highlighting the clinical data, the genomic data. We bring in other kinds of data modalities that increases kind of the complexity of how you search. I think we heard a lot today about the workflow costs and how do we understand them. I was reflecting, I do feel like I've been talking about this for a long time, but I think it's very real now. Like people are on the cloud platforms, they're really using it. And those concepts years ago of how we would monitor and help that, we really need to make that real. It seems like we heard a lot about the portability of the tools and the workflows. And then the fourth one, the light commons metadata model across systems, which I think in a lot of ways is probably linked to the search. Like what are those anchor points, the handholds that you can get, that you can move across the different platforms and then expand your horizons from there. So it's definitely, I think, good echoes across what we heard from today. And, I think there's a lot of good fodder for Monday's sessions where we can get into the details of some more of these and really start mapping out, okay, this is what we want to do in the 6 months. Here's kind of a general roadmap for the 12 months that we'll revisit. And, do we need to either find homes for these unmet needs in the existing working groups, or what's the right space or form to do it? And then I think it was really good to hear some of the new initiatives and other things working on. But I think to the point, there is always that delicate balance between really focusing on what we core needs, what we need to do, and when and where to engage the new opportunities. So definitely interested in hearing everybody's thoughts on Monday. So the Fun Roadshow board will be up and available. I guess if you do come up with a late night, midnight thought, feel free to put it on there. And I think, we can also revisit that on Monday as well. Adam or Valerie, did you want to say any other concluding thoughts before we wrap it up for the day?
No,
I don't think so. There's a lot to digest.
It was beyond awesome. Thanks again for all the speakers and for sort of all the session chairs. I think it provides a really tremendous substrate for Monday and longer term. So thanks, everyone.
Thanks, everyone.
And happy Halloween.
Happy Halloween.
Thanks, everybody.
Thanks, everybody. Have a
Thank
good weekend.
you.
Bye.